So welcome everybody. We are here for our Specialty Day 2021, and it is my pleasure to introduce our president, Dr. Carol Morris, to start the activity of the day. Please, Carol.
Yes, welcome everyone. It's really been quite a year, and I hope you and your families have been safe and healthy. It's really wonderful that so many people registered for this today, and it's great to see our engaged society. I want to acknowledge the executive committee for all their hard work, keeping MSTS moving forward during this time. It's been difficult, and a special thanks to Carrie and Deborah for their hard work this year as well. When the Academy rescheduled their meeting from March to August, several of the other subspecialty societies decided to hold their specialty day virtually in March, and we decided to join them and do the same. It's been a really long time since we all gathered, and staying connected in this way is important, not only for our professional growth, but maintaining strong society bonds. I want to commend the Specialty Day Committee for planning such a fantastic program under such short notice. This concept of advances in musculoskeletal oncology around the world with speakers from across 6 continents is really quite a feat to pull off so quickly. So thank you very much to Nick Bernthal and David King under the leadership of Pietro Ruggieri for all that you've done to make this happen. And thank you to all the presenters today. I'm really looking forward to learning from all of you. Please be on the lookout next week for our spring newsletter coming out with information about the fall meeting that we hope to hold in October. And so just wanted to give people a heads up on that. So without further ado, Pietro, please take over, and we're looking forward to today.
I wish, on the other hand, thanks our president and all the board for supporting us of the committee in this a little bit difficult task because of all the troubles that we've gone through, and also all the need to cut the program. The program we elaborated together with Nick and David. We like that very much, but we had to cut and cut progressively, because when transforming the meeting from an in-presence meeting to a virtual meeting, that is always a problem. But anyhow, I think we could make it, and I have to thank, of course, again, Carrie and Deborah for their continuous support and hard work. And particularly, I am grateful to Nicholas Berntal and David King, because I think that we could work as a team in all the times of the process, and it's been a pleasure for me to work with you both. So before starting, I would really ask Nick and David if they want to add something to say hello or whatever. Please, Nick.
Yeah, thanks, Pietro. It's been a pleasure. And I also want to just take a moment to thank all the speakers, because we've been coming to you guys with cuts as we've been condensing the talk. So thank everyone for being flexible. We know asking you for a 20-minute talk, then a 10-minute talk, then an 8-minute talk is a lot of work. We're really looking forward to it. And Pietro, thanks for leading us.
Thank you, Nick. And of course, I join in these thanks to our speakers and participants. And David, please.
Yeah, no, I echo that. I think it's a real challenge to cover the amount of material you want to cover in a short period of time. So I think we're going to hit the highlights, and certainly now that we're more connected, through email and everything, I think follow-up questions and everything are more than welcome. I'm sure for all the speakers. So enjoyed working with the group, and excited to finally get back together. So thank you.
Thank you, Dave. Thanks a lot. So now it is my pleasure to start the first session on state of research, and I'd like to introduce Mackenzie Tyler, my co-moderator, and she's the one who will start this session on the state of research. Please, Mackenzie.
Thank you, Pietro. It's a pleasure. Thank you all. And I have the great pleasure of introducing our first speaker, which is Dr. Fran Hornacek, who's going to be talking about biomedical research in the USA, which is a broad and exciting topic.
Okay. Everybody see the screen? Okay?
Yeah.
Okay, good. Thank you very much. again, this is I just wanted to touch on a couple things that I think are very important in terms of updates. I have no disclosures relevant to this talk. And I just wanted to comment on, I'll be leaving UCLA to go to Miami, but the UCLA sarcoma team started a long time ago in terms of, multidisciplinary care. And one of the first places where, chemotherapy was used in the treatment of osteosarcoma. And unfortunately, Jeff Eckert has passed, but we remember him greatly. And we were able to raise, thanks to many of you on this call, an endowed a chair in his name, and Dr. Bernthal, congratulate him on being the first person to be in that, awarded that chair. So, as mentioned, we know this is a multidisciplinary approach, and this is in the OR, but also not only in the OR, but also in the clinic as well. We function together as a team, medical oncologists, radiation oncologists, nurses, everybody's very important. And this is just a picture from the MGH OR functioning on a case from, I believe this was a young girl from South Africa that we participated in her care. One of the things that's had a significant impact over the past year or so is COVID, right? And this pandemic has broadly impacted our ability to do research. And I don't know how it's affected your particular academic medical centers, but certainly here at UCLA, there have been a lot of changes and rules applied. And it's actually affected probably the early career researchers more significantly than those that have been doing research for a longer period of time, which I think is something to talk about. And there are actually a number of publications on this as well. This one, talked about the lockdown of research centers, its effect on clinical trials, not only basic research, but translational as well, and as well as clinical trials. It was very interesting about when they did this survey of the basic researchers that only 13% were as productive as before the lockdown. So this has a significant impact, of course, on our research recently. Now, one of the areas that I think has changed dramatically over the past 10 years. So I don't know if Joe Schwab's on the call, but when I was at MGH, I was fortunate to be able to hire the I don't know if that's Nick will take care. I was fortunate to be able to hire his father-in-law, Sodano Ferroni, an immunologist. And when I hired him through the cancer center, I was told that immunology was a thing of the past. And I really shouldn't hire anybody who has anything to do with immunology. Well, that has changed dramatically, as we know. And immunology is really one of the certainly one of the most cutting edge technologies in terms of treating cancer, including sarcoma. Here, Tony Rivas and things has clinical trials on synovial sarcoma. There are a number of clinical trials underway across the world. So there are a number of therapeutic strategies for sarcoma. A number of these involve checkpoint inhibitors, some vaccines, and some like Steve Rosenberg, who is building 11 NCI in things adoptive cell therapy. And CAR T, for example, Sodano Ferroni is interested in CSPG4, and they now are developing CAR T for that. And some of the antigens that are defined in melanoma actually have some applicability to that of sarcoma as well. So just another diagram from that paper, just showing you a cartoon, adoptive therapy, immune checkpoint inhibitors, the vaccines. And some of us have had an ability to work in some of these areas on our own, both in preclinical and in clinical trials in some of this work. And this is what patient Arun Singh, one of the medical oncologists here, that we share. They've had a huge cervical cordoma was treated elsewhere, came to us. We decompressed him, and he developed metastatic disease with a large mass here around in the free chest area, sort of sticking out like a fungating area. He was treated with relatinib and nivolumab, and ended up these the relatlimab is a melanoma targeted LAG target, and the nivolumab is well known as is also used as an inhibitor, works as a checkpoint inhibitor, and this has been around, but it sort of melted the tumor away. So just with 2 immune modulatory agents, we were able to find that this tumor that was not responsible to anything else, including radiation, was responsible to 2 immune therapy agents. So we thought that was significant. There are a number of methods that have been out there, and we know that many of these patients with sarcoma will develop metastatic disease, and a relatively high percentage of them. And how does that develop? A lot of times it develops through drug resistance. So tumor develops a resistance to chemotherapeutic agent. Whether it's acquired or innate, these are things that we can talk about. And over the years, we've been fortunate to develop a number of cell lines that are resistant to chemotherapeutic agents used in sarcomas, including ET743, a trabectedin, which I was fortunate to be one of the first people to work on this with some of the people from Memorial and various other locations around the world. So even with these successful drug agents now, they're frontline agents made for liposarcoma, some of the cells will develop resistance, or you select out from the tumor those cells that are resistant to the agent. There are other immune modulators that use in targeted antibodies. Just here, Keytruda, Xgeva, we know used quite often. And again, adoptive cell therapies, the CD8 T cells, C. Rosenberg, and that's really any SO1 target used in synovial sarcoma. And this has been out there for a while as a target and is found to be quite good. So in general, the clinical trial targets include targeting antibodies, vaccines, adoptive therapy, immunomodulators, and virus therapy. So in terms of targets, these are some of them. CD52, a lot of these may be self-surface markers of proteins or further within the cell and the nuclear region as well. Rank ligand, VGF, has been around for a while, being looked at as a target for some of these. Cancer vaccines, again, NY-ESO1, protein normally produced during birth, also expressed in some of the testes, various other locations. P53, of course, in many of the osteosarcomas is one of the common things along with RB that we find mutated and potential target. Tumor-associated antigens, a number of those, and WT. In terms of adoptive cell therapy, EBV has been used over many years. GD2, HER2, NY-ESO1, again, adoptive therapy. Immunomodulators, these include some of them. And this is where you start looking at various interleukins, as well as blocking or checkpoint inhibitors as well. There are various oncolytic virus therapies which haven't been as successful, and there's also a concern for safety anytime virus therapy is being used, especially at the FDA level. So one of the things that, and how much time, I have 2 minutes left, just we'll end with this is, so one of the areas we worked on is, checkpoint inhibitors. And this is just some of the preclinical work in chordoma as a therapeutic target. We know it's highly expressed in chordoma tissues. Higher expression correlates with metastases and recurrence as opposed to primary tumors. And that's irrelevant depending on whether it's conventional or chondroid chordoma or poorly differentiated. Treatment of chordoma, so we were able to treat chordoma cells with CDK4 inhibitor and affect the growth and proliferation. And some of these data served as preclinical data for translational work in clinical trials. So here you just see CD4 expression, both in cell lines as well as tissues. Some of these cell lines I've been fortunate to be able to expand and now are being used widely across the world, like CH22 that comes from a met. But you can see metastatic tissue is composed compared to UCH1, which comes from a primary, more CDK4 than others, even in the cell lines. Then when you look at the relationship to how the patient's doing, primary, recurrent, or metastatic, and you can see there's a slight, as the level of expression goes up, there's a difference in terms of whether they have mets or not. And you can also, as a potential target, so you can arrest the cells, stop them from going through the cell cycle, you affect some of these checkpoint inhibitors. Here you have CH22 and UCH2 in terms of being treated with checkpoint inhibitors, sort of demonstrating the usefulness probably of, and here you have the checkpoint inhibitor, pelisasimide, that will then, as increasing doses, you get changes correspondingly to the ability to affect decrease in cell growth. So I just want to touch on some of these things. It's a short period of time, but I think two things I wanted to get across was the COVID, and the other thing is immunotherapy and how it really has changed from where we were 10 years ago. Thanks.
Thank you, Francis. Thank you for your wonderful talk. It's really complicated to summarize such important topics like biomedical research in a continent or in a big country in a few minutes, but you were excellent in that. And now it is my pleasure to introduce Professor Panayotis Papagelopoulos, very old friend of mine, who is going to give us a talk on biomedical research in Europe. Please, Panayotis.
Thank you. There. Oops. Sorry. So, good morning from Athens. Thank you, Professor Geri, and I would like to thank also the scientific committee and our president, Dr. Morris, for the invitation to talk on the status of biomedical research in Europe. The aim of biomedical research is to answer medical questions leading to the discovery of treatment, prevention, and diagnosis of diseases. And of course, this is basic, clinical, and translational research from the laboratory bench to the bedside. This is Europe now of the 27 states in 2021 after United Kingdom's exit. One of the main issues in Europe is aging. Life expectancy in Europe is increasing by 2 years every decade, while the United States remains stable. So people in Europe would like to enjoy more years in good health and have personalized treatment available when they develop a disease. Pharmaceutical and biotechnical research and development spending in 2016 to 17 accounted for about 40 billion in biomedical research. Europe's pharmaceutical sector produces about 250 billion in revenue and employs about 750,000 people in highly skilled jobs. The key research and innovation areas include personalized medicine, with the development of new technologies on molecular medicine and genetics, on designs of predictive diagnostic and therapeutic personalized tools, and emerging technologies, artificial intelligence, and big data. One of the problems is antimicrobial resistance, which represents a global threat. Biomedical research aims to improve prevention, diagnostics, treatment, and overcome the reluctance of companies to develop new antibiotics. Biomedical research on rare diseases. The definition of a rare disease is a disease that affects 5 out of 10,000 people in Europe, but rare diseases are common. There are about 6,000 to 8,000 rare diseases affecting more than 30 million European citizens. Biomedical research on environmental factors suffers of underinvestment despite the huge impact on health. The European Research Council is a public body of funding of scientific and technological research within Europe. It has an independent scientific council, which is the governing body, and an executive agency, which is in charge of implementation. Since 2007, the European Research Council has funded more than 5,500 top researchers and over 70,000 postdocs, PhD students, and other staff. More specifically, from 2014 to 2020, the Research Council budget was more than 13 billion, derived from Horizon 2020. 7,000 grantees were funded and 42,000 team members have been supported. Now, in the COVID area from last year, more than 469 million were invested in more than 100 research projects, including clinical trials, vaccine projects, basic research on coronavirus, and other projects. Last year, the European Commission pledged to invest 1 billion from Horizon 2020 into research and innovation to fight COVID-19. This includes 800 million that have been already mobilized and 200 million are pledged. Horizon Europe from 2021 to 2027 is the world's largest multinational research and innovation program of about 100 billion to basic science projects and cross-border research collaborations involving tens of thousands of researchers across the 27 European states and other countries. The 2021 to 2027 budget of the European Research Council will be more than 16 billion as a part of the Horizon Europe's research and innovation program. In this new program, the health innovations represent a main area of research, and more specifically, a key mission area includes cancer research. Malignant neoplasms are the second cause of death in Europe. The impact of biomedical research on the European society is reflected in the success of modern medicine, including the decreased death rates for all diseases except the nervous system disorders. In 2017, European Union spent more than 320 billion on research and development. Research and development investment in Europe in 2017 was about the 2% of the GDP, lower than Japan and the United States, while it was about the same level as China. However, Europe's 2020 target for research and development expenditure was increased to 3% of the GDP. Today, the pharmaceutical research and development expenditure in Europe is about the 2/3 of the United States. Europe's pharmaceutical and biotechnical industry has for many years been the world's principal developer for new medications. However, since the 1990s, this position has been taken by the United States. And between 2014 and 2018, about half of the new chemical and biological entities in the world were developed in Europe. The projected global biotechnology market by 2026 will be about $742 billion, and Europe will hold about the 40% of the global biotechnology market share. Of the top 10 biotechnology companies in the world, for 6, the mother company is in the United States, and for 4 in Europe. United States, Europe, Japan, and China are the world's 4 most important producers of science. As we see here, Europe is well balanced in terms of specialties with greater strength in medical research. Europe produces 35 to 40% of the world's scientific publications, and 8 out of the 10 top publications are on biomedical research. Jointly, Europe and the United States produce almost the 2/3 of the world's total publications in the field of biomedical research. Now, the basic orthopedic research has been expanding the last decade in many fields. A recent study reviewed all the publications, the orthopedic publications from 2010 to 2014 in the 15 top orthopedic journals, according to the impact factor. There were about 23,000 publications, and Europe had a similar number of publications and impact points as the United States. Among the European countries, UK had the highest number of orthopedic publications, followed by Germany, Italy, and other European countries. There are many orthopedic research departments and laboratories in Europe involved in all fields of orthopedic research that we don't have the time to analyze, of course, now in detail. There is the European Orthopedic Research Society promotes the research and development in orthopedic surgery in Europe. The European Society has an annual meeting and 2 affiliated open access journals for European research publications. Also, EFORT supports highly quality research projects in orthopedics and traumatology in collaboration with the national and specialty societies, as well as with other partners in Europe. This is the European Orthopedic Research Network with an online interactive map of all the research projects in Europe and collaborations. Finally, the European Musculoskeletal Oncology Society supports many multi-center clinical studies in musculoskeletal oncology. So I think that it would be appropriate to finish this talk devoted to the European biomedical research and discovery with a reference to the penultimate European Exploration Odyssey, which keeps remind us that the journey of discovery is as important as the destination. Thank you for your attention.
Thank you. That was absolutely wonderful. A really comprehensive discussion there. I'm going to present Dr. Tutsia, who's now going to speak about biomedical research in Asia. I'm very much looking forward to this, and I've enjoyed spending time at Kanazawa with Dr. Tutsia, who's an absolutely phenomenal surgeon and educator.
Thank you, Wakenda. Good morning from Kanazawa, Japan. It's a great honor for me to join here. I would like to talk about biomedical research of musculoskeletal oncology in Asia. Oh, it doesn't work. How do I proceed? I have no disclosure. Asia is the largest and most populous continent. Its 4.5 billion people constitute roughly 60% of the world's population. This presentation focuses on East and Southeast Asia. I sent questionnaires to Asian hospitals in addition to Japan, Malaysia, Thailand, Philippines, China, and Taiwan. First question is, are there nationwide clinical study groups in terms of musculoskeletal oncology in your country? Except for Malaysia and Taiwan, they have the clinical study groups. In Taiwan, first biggest tumor hospital is doing everything. Philippines MSTS, Thai MSTS. And second question is it possible in a country to conduct multi-center randomized trial for musculoskeletal tumors? All countries answered yes. Third question is, do you make a list of prospective multi-center studies in a country in the past 5 years? Liposomal doxorubicin for osteosarcoma treatment, single-arm multi-center prospective clinical study of anlotinib and doxorubicin combined with PD-1 and ifosfamide in the treatment of soft tissue sarcoma, proximal humeral prosthetic reconstruction, and so on. Tumor-bearing recycled autograft, such as frozen bone, pasteurized bone, and irradiated bone are popular in Asia. Fourth question is, what's the ratio of prosthesis and biological reconstruction? In Japan, 70% prosthesis, 30% biological reconstruction. In China, almost same. In Thailand, completely opposite, and 70% biological. In Philippines, 55% still amputation, 30% prosthesis, 15% biological. Malaysia, 50-50. In Taiwan, similar to Japan. I show one representative case, 10-year-old boy with osteosarcoma in the right distal femur. Preoperative chemotherapy was very effective. We planned joint preserving excision and reconstruction with a frozen autograft treated by liquid nitrogen. This is the excised specimen and the radiograph after tumor excision. After freezing and thawing, we performed reconstruction using double locking plates. Radiographs immediately after operation, right side radiographs 5 years after operation. You can see excellent bony union, no complications. 5 years after operation, we conducted tibial lengthening to compensate 4 centimeter limb length discrepancy. This is the radiograph 8 years after first operation. The recent limb function, you can enjoy soccer, football like that. Now, we are doing research to review the mechanism of revitalization of a frozen bone and to promote revitalization using adipose tissue-derived stem cell seed. Revitalization of frozen bone is promoted significantly. I introduce each group activities together with EMOG, East Asia Musculoskeletal Oncology Group. First, JCOG. JCOG is a multi-center clinical study group for cancer treatment, fully funded by the National Research Grant in Japan. The goal of the JCOG is to establish effective standard treatment for various types of malignant tumors by conducting nationwide multi-center clinical trials, including chemotherapy and surgery. There are 3 ongoing trials for musculoskeletal tumor field. For example, a randomized phase 2 trial of second-line treatment for advanced soft tissue sarcoma comparing trabectedin, eribulin, and pazopanib. We have 3 treatment arms. Primary outcome is progression-free survival, and the secondary outcome shown in slides. A randomized phase 3 study of denosumab before curettage for Janssen tumor of bone is also ongoing. Representative publications from JCOG about high-grade soft tissue sarcomas and Janssen tumor of bone. Next, JMOG, Japanese Musculoskeletal Oncology Group, is also active for doing research. One of the best papers from JMOG published in 2002, Journal of Clinical Oncology regarding osteosarcoma lung metastasis. Publication from JMOG. JMOG is basically conducting retrospective studies. Next, I introduce EMOG, East Asia Musculoskeletal Oncology Group. With more than 80 members from many countries, EMOG is a genuinely open research group in the field of musculoskeletal oncology. First meeting of EMOG was 2004 in Korea. The latest meeting was held in Athens 2019 during ISO's meeting. There are ongoing projects of EMOG regarding complications of recycled autograft, functional scoring, and PVNS. Publications from EMOG with regard to post-radiation sarcoma, total scapular excision, osteosarcoma with pathological fracture, osteosarcoma in Asia, and prognostic factors of recurrent Janssen tumor. China has the first largest population. The number of clinical trials is increasing dramatically. In particular, clinical trial of immunotherapy are dramatically increasing even for sarcoma. China is doing a lot of phase 1 and phase 2 clinical trials, whereas United States has the largest number of preclinical agents. The number of phase 1-2 clinical trial is a key to release new drugs on the market. How about Japan? Using iPS cells derived from sarcoma, oncogenesis of EWS-FLY1 was demonstrated, published by Stem Cell Reports in 2016. Also, limited availability of T-cell immunotherapy will be solved by means of iPS cells application, also published by Stem Cell in 2018. In conclusion, multi-center and multinational clinical studies are going common in Asian countries. And there are also unique Asian studies, such as tumor-bearing recycled autograft. Basic research and clinical trials will increase in the future with the advantages of large population, economy, and basic technology. Thank you. Thank you for your attention.
Thank you, Hiroyuki. We thank Professor Tsuchiya for this excellent talk. It's not so easy to summarize all that's going on in the biggest continent ever, so Asia, but it was good in that. And now it is my pleasure to introduce the last presenter of this first session on state of research, who is my friend, Professor John Healy. He will give a talk, a lecture on what's new in basic science in musculoskeletal oncology. Please, John. Is John here?
Don't see
I don't
John
see.
here.
Do you know if Carrie or Deborah, do you know if Professor John Healy is here with us or not?
Somebody call
I do not see him.
So why don't we go to questions?
him? Should we go to questions? If he's, yeah. Wait, I don't know. Do you have a-
Why don't we go to the questions for the first three speakers?
I agree.
I'll
I
try
agree.
and,
I agree.
okay.
It's a good idea. Then we check if John is here now. Please, Joaquinda, do you want to
Yes.
start?
I will. So I have a question from Kurt Weiss, who's asking Dr. Hornacek about whether he has any patients specifically on the SARC 032 trial
Yes.
with preoperative radiation and prednisone, and what are your thoughts about this trial?
Yeah. So that trial was thought about a while back, actually through SARC. SARC, as I hope most of you know, is the clinical trials group that we actually had a spore couple, I think 2 spores through SARC, and the only other spore grant was through Memorial, but that did not involve kids. But one of the projects had to do with this particular trial of a checkpoint inhibitor, Keytruda or Pembro plus radiation. And Dave Kirsch is the one who was in charge of that project. So actually, after we saw the question, we contacted our radiation oncologist who's in charge of that at our facility here at UCLA, and Nick got an update about that. So, but it is so it's a checkpoint inhibitor plus or minus radiation, soft tissue sarcomas greater than 5 centimeters.
Yeah, I think, Kurt, our experience has been somewhat limited. We had 4 patients on the trial and 3 got randomized to not receive the drug. So, it's not a blinded study, but we've only had 1 patient who received both the Pembro and the XRT. So not sure we can speak with any, our sample size is not enough to say anything.
There are a number of institutions across the United States involved, including Memorial, Michigan, Pittsburgh, including your place, Kurt. And I think the trial is going to last until, I want to say, It's still going to go on until 24, 25. So it's not something that's going to stop right away.
Thank you.
I don't see other questions. Do you want to ask other questions, or we move to the second lecture, Joaquinda?
You know, I have some personal questions, but I think these can wait. They're not dramatic questions, so we could potentially move to the next lectures and then come back.
Okay. So Panayotis, from the highest level of the Parthenon, where I see you, we heard from you about all the advantages and problems of the economical distribution and activity encouraging research and biomedical research in Europe. But what is the most critical contribution in orthopedic oncology from the recent times from Europe, in your opinion, is one question. And the other question is, if you could see that in Europe, there's a equanimous distribution from the different countries in the contribution to the, and if this can be really mediated by our European society, which is a prominent role, the EMSAS.
Correct. Yeah, I didn't want it to take a time because my talk has been shortened, and also to take time from the lecture that the president of European Musculoskeletal Society, Andreas Lennart, has later today. There are many multi-center, as I said, clinical studies involving musculoskeletal oncology through the society right now. Some of them similar that with studies that already have been referred by the previous speakers, and with this collaboration, many studies have been already published in different subjects like sarcoma during pregnancy or extra-abdominal fibromatosis multi-center study that with new non-surgical treatments, a study about clear cell chondrosarcoma. So I think that the president of our society will discuss this in his talk. So this is why I didn't want to expand my lecture on these studies on musculoskeletal oncology. And just I gave you an overview only about what's going on in biomedical research in Europe. And I think that more specifically regarding the musculoskeletal oncology, our president will give his talk later today.
Thank you, Panayotis. Joaquinda, do you want to add some on this?
Yes. Well, I have a question for Professor Tutia. I was really interested in the numbers that you presented in terms of the differences in regions on prosthetic use versus biological reconstructions or using not prosthetic use. And I saw the difference with China and Japan having mostly prosthesis versus a place like Thailand that's doing the very opposite. Do you think that that's related to resources and availability within each individual country? Is there more availability of prosthetic use in Japan, for instance, and in China than in Thailand? And is that playing a role, or do you think it's more of just a regional difference in cultural differences?
Yeah, thank you very much for your question. Probably it depends on the availability of prosthesis and allograft. Also, religion is also one factor. In Thailand, most of Thai people, they like biological reconstruction. Also Japanese. Yeah, yes.
Okay. Interesting.
I think John is ready now. I saw John. Welcome, John. I hope the technical issues are fixed now. Not really.
No, but
There's an echo. There's an echo.
so I've been here on the whole meeting, but it's taken 4 different entries. So I apologize to everyone for that. And supposedly my slides are being presented through the MSTS, which I submitted, and if they don't have them, I can pull them up again.
We
But
will,
that
they should have
should
it.
have been
Deborah,
prearranged.
Carrie,
So
can you tell us
is that
something?
available or not?
Do we have the slides from Dr. Ely recorded? Yes.
No, I don't know if they can.
Yes, they're here.
Oh, great. Okay, so thanks for the invitation, Dr. Morris, Dr. Ruggiero, and Dr. Tyler. And so these are supposed to be advancing automatically, but that doesn't seem to happen.
Terry may have to advance for you.
Okay, so I'm going to be discussing advances in basic science. I look forward to meeting in person with all the new people in the field and my old friends. My disclosures are here, none of relevant to the group. I'll be covering the whole field of orthopedic oncology and have selected a few to focus in these next 8 minutes. And basically, I'm going to provide you my top 10 for this year. There are things of interest to me that I think are relevant and moving the field forward. But it's a subjective thing, of course. So everyone would have a different list, perhaps. Advance. So number 10, next, is the identification of osteosarcoma in a dinosaur. And so this has been found in mammals, but this is the first indication of this disease in reptiles, and I think gives us some new information regarding the pathophysiology of this disease. Next. And number 9, metabonomics are always enticing. You know, patients want to know, is there a blood test for my disease? And this is an interesting paper where it has been identified with a serum test, identifying 6 genes linked to cholesterol metabolism that highlight the presence of the disease and, in fact, prognostication. And so it's worth looking at. Next. Injury has always been a concern, and does that cause cancer? And here's really the first mechanistic description of this. It's in pancreas cancer, but I think it applies throughout, where injury, then working through KRAS, and the lack of epigenetic repair mechanisms has led to pancreas cancer. And I think this highlights really the mechanistic aspects of how injury can be correlated with our diseases. Next. 7. Genetics. There are several things I'm going to lump into this, but the first one is the identification of proper cell lines for use, in this case in all cancers, defining their metastatic potential. But as you note in the box on the picture, this also includes those in bone and soft tissue sarcoma. So if you're doing basic research and are interested in metastasis, you need to look up this new data, and it's worthwhile. Next. Dating back from the Human Genome Project, there's been a revolution since the year 2000. And in the right upper graph, you can see how the principal genes that we've been interested in have played out in a number of publications, and P53 being the supreme one. But if you also look here on the left bottom graph, the orange ones, the number of non-coding areas that are the subject of investigation are increasing dramatically, way ahead of other genes. And in fact, 90% of the proteome is now not being targeted by drug companies in pharmacology, and provide a great opportunity for new advances beyond what we see in genes themselves, coding areas. Next. And even in the perennial P53 kingpin, there are differences in understanding its use. And here in work in the gut, you can see how it's an oncogenic gene promoting work in the Wnt pathway. But after antibiotic use, altering the biome, it totally reverses the effect of P53 and becomes a tumor suppressor gene once again, even mutant disease. So work on the metabolome and now the microbiome are very important in how these genes work and are worthy of our attention. Next. 6. Image-guided drug delivery is a very hot topic still and very sexy, and ultimate advances in what are called the particle and particle PIP mechanism are important, and gold as a carrier and imager is extremely of interest now, allowing imaging with CT and potential modification, for example, with hyperthermia applications. And so these PIPs are very good. Next. And they're utilizing nanoparticles, which are basically in the 10 to 100 nanometer range. They avoid complement, renal filtration, the reticuloendothelial system, and concentrate by a variety of mechanisms passively in the tumor. They're easy to manufacture and sterilize, but not to scale. So that's been holding back this work. Next. But there has been progress, and I urge everybody to take a look at this great review published this past year, looking at the different kinds of nanoparticles and highlighting that there are 9 active clinical trials using nanoparticles right now. So they are breaking into clinical practice. Next. 5. In the reconstructive domain, 3D printing has been of great interest in marketing, really. But this is the first report of actual retrieval analysis of 3D printed implants and demonstrating proper bony ingrowth and highlighting the biocompatibility of these with better elasticity and matching of bone qualities and improving over other alternative reconstructive implants. Next. Number 4. The cumulative mutational burden is extremely important. And in this work, it highlights how the total mutational burden in osteosarcoma has a twofold difference in the mean progression-free survival and the overall survival, where remarkably high mutational burden has a better survival than the lower one. So this is a little bit curious, and understanding this may have some implication in terms of the immunotherapy opportunities that Dr. Hornacek discussed earlier today. But anyway, it's a marker for prognosis in osteosarcoma. Next. Number 3. One of the oldest materials that's available, silk, actually has had a great resurgence in its utility and is worthy of much further investigation. And in fact, the fibroin from silk can now be clinically manufactured, and so not just depend on silkworms for product production. And so it's wonderful. It's biocompatible, biodegradable, absorbable, low immunogenicity, and it can be tuned to its mechanical properties. And so it's going to be a great advent for soft tissue reconstruction, which is one of our big problems. Next. And it's really, the fibers are tougher than other synthetic fibers. They have the highest strength among natural fibers. They can cross-link and intercorporate with the 3D bioprinting and actually look for not just soft tissue reconstruction, but attachment to these reconstructive implants that we would like for surgical use. Next. And it ultimately can produce a variety of scaffolds that can integrate well, and examples of that are shown in the bottom left and right pictures. Next. Number 2 is basically a deep learning and artificial intelligence. And so the #1 highest cited journal reference in 2020 was from Nature, originally in 2015, where they used a novel deep Q network to basically surpass anything that the human mind can do in any gaming, and beating the world Go champion. And so this now is the most highly cited reference, and has produced 13 major papers in Science and Nature, and 3 so far here in 2021. So this is taking over. How it's going to impact in orthopedic oncology remains to be seen. It requires very high quality data to use, and so unfortunately our data may not be up to snuff, but there's great potential here to really remake our whole field. Next. And #1 for the year in research is coronavirus, and this has multiple manifestations. Next. We heard about the European budget, but here just in the UK, for example, documented in Lancet Oncology, they had to cut 45 million pounds from the UK research budget, and there's been a diversion of funds into other fields. And I highlighted this last year in the MSTS editorial from the subject, but it's totally changing how we think of ourselves and our field and interaction with research, but we need to keep going. Next. There's some interesting things, however, that have come from this research, and I've highlighted 2 of them here. One is that the Neanderthal gene actually predisposes you to get coronavirus and to die from coronavirus. And so that's a curious and fascinating thing, the cause of which is uncertain. And that dogs are actually able to smell it and diagnose it. And so if you don't have other tests available, and you don't have our Governor Cuomo giving you access to tests that no one else can get, you can get your dog to maybe help out in that regard. Next, please. But it's impacted on education, our ability to interact internationally and have either even domestic, but certainly foreign fellows, and your ability to recruit people now with Zoom interviews, and you have technical problems just like I had this morning. I apologize again, but this impacts on how we prepare for the next generation. Next, please. And finally, postscript, and this is a focus from the journal Nature, where their editorials and correspondence have highlighted the importance of enhancing equity throughout the research as well as other domains, and dating back all the way to Rosalind Franklin, highlighting her work, and the recent call for improved participation and opportunity for women and underrepresented minorities throughout all of our research endeavors. And I think it's worthy of all of our attention to be with the editors of Nature and try to advance this aspect. So with that, I thank you, and I apologize again for my technologic problems earlier.
Thank you, John. Thank you. Very interesting talk, really remarkable. I see that there is a question now. I don't see that anymore.
Well, I saw one. Dr. Westruck asked or said that she likes these Zoom interviews, and there are aspects to it that certainly are good, and it's economic and effective for the applicants in certain respects, but they don't get to see the program. We don't get to see them in person, and the social interaction remains deficient. But you know, they're very costly, both in time, disruption of the programs where people are coming from, and our time as well. So there are some advantages to that, just as there can be with telemedicine in established patients, but new patients are really hard to do. And I think that applies to interviews.
Thank you, Rosanna, for your question. No, I was focusing on this other question from Robert Henshaw. He's asking, John, any thoughts about mRNA therapy for sarcoma now that there is a proven delivery mechanism in COVID vaccines? So the question is about mRNA therapy for sarcomas. Any research on this, any opening or any feeling from your standpoint?
You've got me there. I haven't encountered anything in that, and I you know, searched in the top 10 research journals specifically in trying to prepare for this talk beyond my own regular reading, and so I'm not aware of it, or through my institution we don't have any program in that. But it's a fascinating idea, quite possibly, but I really don't know. It's a great suggestion, though.
Were they referring to microRNAs, or what were they? I don't know if they were. You know, I mean, a lot of those changes are common in different cancers as well. So they span cancers. A lot of changes in certain ones that you find, whether they're up or down regulated, you find them across many different cancers. But we thought were unique to, say, a sarcoma is not.
Right. Thank you.
So.
I wanted to ask a question to my friend Lorenzo Lorandel. Hi, Lor. I'm reading what you said about that it's been announced that the UCD Ortho received the first federal funding for a US Ortho department to look at molecular targets in COVID. So although it's a little bit out from the orthopedic, but we are all interested in this, I'm asking you, besides congratulating with this, I'm asking you if you can say a few words about this study of molecular targets.
Sure. So Dominic Hodenschild, who's in our lab, has been looking at post-traumatic osteoarthritis and cyclin-dependent kinase pathways. And he has been able to find he's developed some small molecule inhibitors of CDK9, and the CDK9 pathway in COVID is out of control, like many. And so he got a couple of federal grants looking at targeting CDK9 pathways in musculoskeletal tissues of patients afflicted with COVID, but then also we're working with internal medicine for the pulmonary fibrosis associated with COVID. So we've got a bunch of different stakeholders, and we're pretty excited about it.
Thank you, Laura. Please, Vakenda, do you want to close the session? And
thank you all. That was a really outstanding, thank you, Pietro. That was a really outstanding session, a really wonderful discussion. And I thank you all for all the panelists for being involved. And we'll move on to our next session. And I'll let Dr.
Thank you.
Great. All right. So we're going to move on to critical issues and updates on emerging drugs. Rosanna is going to start us off. And Rosanna, if you can keep your talk to about a minute, I think we can catch up, everybody. So appreciate that.
Okay, can everyone hear me? So thank you for the opportunity. I have no financial disclosures, but I should disclose that I myself have never brought a drug to market. So I learned a lot preparing for this talk, and I hope to share that with you. So I'd like to go over the general FDA approval process and then 2 incentive programs to get sponsors to really invest in rare diseases, and that is the Orphan Drug Act and the Rare Pediatric Disease Voucher Program. And then I'd like to close with having us think about how we prescribe drugs and how we think about cost effectiveness. So the drug discovery is is a very long and very costly process. Only a few compounds will actually make it to market of the over 5,000 compounds in active drug discovery. Once a drug actually makes it to clinical trials, it is a little bit more likely to get to market, maybe 1 in 10, 1 in 5. But overall, it takes about 8 to 12 years on average for a company to bring a drug to market, and it can cost between $1 and $2 billion. So as you can imagine, there's very little incentive for a company to invest in a rare disease that has a small market, and they'd be unlikely to recoup the cost of development. Another barrier to novel treatment development for rare diseases is the fact that unlike diseases that affect a large portion of the population, the NIH is not doing a lot of work on natural history or mechanistic research in these rare diseases. So that research all needs to be done by the sponsor, thus increasing the time to bring that drug to market and making it more costly. So what is a rare disease? Well, a rare disease is defined as a disease or condition that affects fewer than 200,000 people in the US. So while each disease may be rare, rare diseases are actually quite common. So 7,000 diseases or conditions have been identified as a rare disease or disorder. And this affects 30 million people in the US and estimated to affect 130 million people worldwide. Half of these individuals are children. And unfortunately, of these 7,000 conditions, effective treatment's only available for about 5%. So there's a huge need to develop more treatment for rare diseases. In 1983, Congress passed something called the Orphan Drug Act. And it incentivizes the development of drugs for rare diseases by offering a 7-year marketing exclusivity to the sponsors of an orphan drug. And this is compared to the usual 5 years. It also offers a 50% tax credit for the research and development cost. So this graph here on the right demonstrates that this Orphan Drug Act has been largely successful. In 1984, only 3 drugs came to market under the Orphan Drug Act. And by 2014, 49 new drugs were approved by the FDA under the Orphan Drug Act. And as of 2018, there's over 730 drugs available to treat rare diseases brought to market under the Orphan Drug Act. And interesting for us, over half of these drugs are directed for oncologic conditions. The other incentive program is called the Priority Disease Voucher Program. So this was actually started in 2006 to address the need to develop drugs to treat neglected tropical diseases like malaria. In 2012, it was expanded to include rare pediatric diseases. Rare pediatric diseases are defined as a condition affecting less than 200,000 children between 0 and 18 in the US. So this is really interesting. And the way this works is, let's say I'm a company and I want to develop a drug for synovial sarcoma. Given that it's a rare pediatric disease or Ewing sarcoma, I can say, I can apply for this voucher. And it costs between $2 and $5 million to get a voucher. And what that will do is I can use that voucher on the next drug in my pipeline. And the FDA will then review and issue a decision on my next drug within 6 months. And I can bring that next drug to market quicker. So this is helpful if my next drug is going to treat cholesterol, and I think it's going to be a blockbuster drug. Now I can recoup the costs that went into developing the drug for synovial sarcoma by getting my cholesterol drug out there earlier. Another way this incentivizes smaller companies is these vouchers can be sold to another company. So if you have a small company that's only working on one product for a rare disease, they can get a rare disease voucher and then sell it to a bigger company. For instance, in 2017, Sarepta Pharmaceuticals developed a drug for muscular dystrophy. They obtained a voucher. They sold it to Gilead for $125 million. Gilead then used that voucher to bring an HIV drug to market quicker. So it may take a little bit of time before we know if the Rare Pediatric Disease Voucher Program is doing what it's intended to do, in that we have more drugs available to treat these diseases. But Huang and colleagues compared drugs that were intended for adult rare diseases versus pediatric rare diseases. And they found that those drugs aimed at pediatric rare diseases did progress more quickly through all phases of clinical testing and were more likely to be first in class. There also was a higher rate of progressing from phase 1 to phase 2 clinical trials. It's important to note that this program is not going to allow a company to bring a bad drug to market. The only guarantee is that the FDA will review the drug quicker and they can bring a good drug to market more quickly. So shifting gears, we're hoping that these different programs result in more novel treatments for rare diseases and maybe more diversity in the drugs that are available. But what do we do when there's multiple drugs that treat similar diseases? And I think a good example for us is treatment of metastatic carcinoma to the bone. So we know that both denosumab and zoledronic acid are effective at reducing pain and skeletally related events in patients with metastatic carcinoma to bone. Some studies do show that denosumab is a little bit more effective by showing a delayed time to first skeletally related event compared to zoledronic acid by 18%, but zoledronic acid is still quite effective. So which one would you prescribe? So denosumab costs about $1,828 per dose, whereas zoledronic acid only costs $140 per dose. The administration fee is about the same. Shapiro and his colleagues performed a cost-effective study looking at the difference between denosumab and zoledronic acid in treating skeletally related events for patients with metastatic carcinoma to bone. They first found that the quality adjusted life years were essentially equivalent between monthly denosumab, monthly zoledronic acid, or quarterly zoledronic acid. They then found that monthly denosumab was 9 times more expensive than quarterly zoledronic acid. And compared to quarterly zoledronic acid, the incremental cost per mean skeletally related event avoided for denosumab ranged between $160,000 to $350,000. So why the difference? Well, mostly due to patents. So the denosumab patent will expire in 2025, whereas zoledronic acid has been available in generic form since 2013. And as our esteemed colleague would say, orthopedic oncologists should not be led by pharmaceutical industry. We should own the field. So that is mostly food for thought. So I really ran through this talk quicker and I cut out a ton of slides, but I'll just press on. So despite the high cost of bringing new drugs to market, there are incentives out there to develop drugs for rare diseases affecting the musculoskeletal system. The Orphan Drug Act, which has been around since 1983, and the Rare Pediatric Disease Voucher Program both seem to be affecting the availability of drugs for our patients in a positive way. There are more novel therapies available for our patients than there were 10 and 20 years ago. However, even these incentive programs, there's still a very high cost of development for these drugs, and this cost is mostly borne by the healthcare system and ultimately the patient. We as prescribers should be able to evaluate available cost-effectiveness data, and this will guide our prescribing practices and hopefully lower overall cost. Thank you.
Great, thank you. Thank you, Rosanna. Nice talk. I know that our pharmacy or our institution is starting to try and get that data up front and center for us as far as the cost of everything we use. So I think that's going to change behavior, certainly. Great talk. Our next talk is going to be by Dave Greenberg. He's going to talk about bisphosphonates, denosumab, and beyond. Dave?
Good morning. I'd like to thank everyone and the organizers for inviting me to talk this morning. The title, as Dr. King talked about, is Bisphosphonates, Denosumab, and Beyond. I do not have any disclosures today relevant to this talk. Just as a little bit of background, most of the osteolytic bone tumors that we are familiar with and we treat on a daily basis do have a common mechanism of supernormal osteoclastogenesis. Typically, the rank ligand pathway is involved in that, and that is what leads to the clinically seen bone destruction that we often are dealing with. As we delve deeper, we've heard about pathways today, and it's somewhat exciting that, in these tumors, as we're studying them and getting to know their mechanisms and the underlying pathways, we are starting to identify driving mutations, such as the H3F3A gene, which is a driver mutation in giant cell tumor bone. these mutations can serve as both diagnostic and therapeutic targets, which we'll touch on briefly. So both bisphosphonates and denosumab, through different mechanisms of action, target the rank ligand and rank pathway. And by targeting that pathway, they do decrease the osteoclast activity and thus help control some of that osteolytic process that we are seeing and dealing with. So looking specifically at giant cell tumor of bone, we know that with oral or intravenous bisphosphonates, you can decrease local recurrence, you can stabilize recurrent disease. Systemic denosumab has been thrown to not only stabilize disease, but help patients relieve their pain. And we're starting to see that bone formation at the periphery, which can convert a potential resection into an intralesional treatment. So for example, here's a patient who had distal radius giant cell tumor. You see here prior to neoadjuvant denosumab, then after several cycles of denosumab, you see the bone formation at the periphery. Histologically, you do not see any active giant cells, and you see the quiescent stromal cells. So systemic therapy can have a benefit, but there's some concerns. That includes the AVN of the jaw, the atypical femur fractures, the rebound hypercalcemia. The duration and timing is controversial and has become more so with some newer articles that have come out. And that new bone formation at the periphery that I just showed in that one radiograph can actually cause intralesional therapy to be more challenging, possibly prone to local recurrence. There's some fairly recent articles that have come out that started to describe a potential increased local recurrence with the neoadjuvant systemic therapy in denosumab. A fairly recent review article published several of these articles, and if you look at these two columns here, you have local recurrence with denosumab plus curettage versus curettage alone, and you're seeing an increased rate of local recurrence described with the neoadjuvant denosumab. The bottom line is the data today is somewhat controversial. There may be prone to local recurrence, but it's certainly not clear. A recent Lancet Oncology review of 500 plus patients did look at the risk benefit and did find that denosumab therapy remains favorable. Some of the other concerns discussed is the idea of progression after you stop taking the therapy or persistence because the neoplastic stromal cells are quiescent. There is some that we do get apoptosis with bisphosphonates, which is somewhat different, but still we're not sure if you don't get this recurrence after cessation. There's an ongoing study looking at the idea of a reduced dose density with maintenance therapy. That data is not published yet, but there is some thought of that may be one way around these issues. And then a topic that we've talked about at our previous meetings is sarcomatous transformation. It's important to understand that this is not proven. There's certainly no causal proven mechanism. Immunosuppression maybe some think is an etiology. It's a fairly rare report, but it is something we have to at least keep an eye on and consider. One side note, with H3F3A mutation being a diagnostic agent, we can help utilize that to distinguish between the sarcoma and recurrent giant cell tumor. So switching gears to the idea of local therapy. It's been well published about traditional adjuvants. None is clearly shown to be superior. Some question whether it's needed at all if you do a good job with the local treatment. Dr. Gerder and her colleagues published a very good meta-analysis that really found local recurrence to be similar with or without intralesional, or with or without a traditional adjuvant. Recent current concepts looked at the recommendations and the data. They found that no single agent was superior, and that meticulous curettage is the most important factor. So with traditional adjuvants not being clear, systemic therapy having some concerns, that opens up the idea of the local biologic adjuvant. This is not a new concept. Dr. Healy and others have talked about it in the past decades ago about using local biology as a potential target and therapeutic mechanism. Dr. Lee and I published our data on the use of bisphosphonate-loaded bone cement. It was a feasibility study. We showed a recurrence rate of 5.9% in our small study, showing that it's potentially safe and effective, which has led to the ongoing giant randomized trial that we are conducting, which is a multi-center looking at bisphosphonate bone cement compared to non-BP loaded bone cement with regards to local recurrence for giant cell tumor. Many on the meeting today are involved. There's 13 centers and more upcoming. It's actively recruiting, it's RAF sponsored, and it's hopefully going to help clarify the idea of the biologics. So switching to aneurysmal bone cyst, another tumor that many of us treat. So similar to giant cell tumor, there's been different protocols utilized, but the bottom line is denosumab is used for to treat ABC. It's been published in several case series. It's similar patient selection, axial or unresectable disease, recurrent disease, both neoadjuvant and the adjuvant role. The early data is favorable, but again, there's some concerns, especially the rebound hypercalcemia that's been seen in the pediatric population. Denosumab is not the only biologic agent that's been used. And again, it brings up, I think one of the themes of today's meeting is the idea of the understanding the pathways and the biology. Doxycycline has been published. Dr. Myerson and his group looked at that, and that's the MMP pathway. They've shown some early data that's pretty exciting. Anecdotally, I've seen some good results with doxycycline. We know that there's sclerotherapy that's out there for these ABCs. The challenge remains, though, we're not sure which of these is the best. Maybe none of them are, maybe similar to the meta-analysis that was done by Dr. Gerder and her group, maybe really none of them matter, maybe it does. I think really one of the key point I'd like to highlight today from my talk is that, you know, what we've done is we've taken the basic science, we've taken the clinical research, the clinical data, we're doing the randomized trials, and hopefully we're going to combine that with the upcoming tumor registry, and we'll be able to figure out which of these pathways, which of these targets can really help affect the treatments, such as we started to see early with the giant cell tumor and the bisphosphonates, understanding the rank ligand pathway. And with that, I thank everyone for their attention.
Appreciate that, Dave. Nice talk. Certainly a lot to cover in a short time. Thank you. Our next talk is going to be on CSF1 receptor blockers, failures, successes, and side effects by Brooke Crawford.
Good morning, everybody. Everyone can hear me okay?
Yep.
Okay, good. So I don't have any disclosures. As we all know, CSF1 receptors, kind of the milieu of the tumor tends to be CSF1, especially in TGCT, tends to be more towards this immunosuppression and tumor promotion pathway. So the macrophage M2 category. And so that's why it became a target for solid tumors in general. TGCT is pretty unique in that it is very, very CSF1 positive. So that's why, that's sort of the mechanism behind the whole thing for those of you who haven't looked at it in a while. As a monotherapy in solid tumors, we haven't seen overwhelming responses with our CSF1 receptor blockers. However, we're starting to recognize that CSF1 increases with systemic therapies and radiation. And so perhaps using it as an adjuvant might be in the future, but as a monotherapy in most solid tumors, we haven't had a ton of success. When you start to look at TGCT, you know, imatinib, Gleevec was sort of the starting point with this. Some small studies showed about a 1/5 response rate in patients to Gleevec, and its overall response. So again, not overwhelming, but at least something sort of stable. And then when we started looking at multi-institutional retrospective data, about 31% of patients had a defined RECIST response rate, but getting close to 80 approached systemic improvement. And these responses were durable at 1 and 2 years and started to drop off after 5 years, but a pretty durable response for TGCT to Gleevec. The adverse effects were mostly grade 1 and 2. Grade 3, though, did occur. We saw some hepatitis, spatial edema, neutropenia, and fatigue. As we started getting more specific in our tyrosine kinase inhibition to CSF1 inhibition, we looked at EMAC and also PEXI, so a monoclonal antibody and a small molecule inhibitor, and had some pretty good results actually with this. Although the patient cohorts were small, the response rates were durable in a year. For a year in PEXI and almost up to 2 years in EMAC. So this led to the Enliven trial for pexidartinib, which was a randomized control study with PEXI versus placebo. It lasted, the treatment arm lasted a total of 24 weeks, and then there was an open label extension. Our overall response rate for RECIST was 39% at that 24-week treatment mark. And then the tumor volume score was actually 56%, so the actual volume of the tumor. And so this was pretty encouraging for everyone involved in the study. COVID sort of led to the ability for us to have long-term data for this talk, as I was scheduled to give it last year. And this data came out in October. After 39 months of a median of 39 months of treatment, the overall response rate from RECIST actually went up to 60% from that 39%, and the tumor volume score increased to 65%. What was really kind of exciting was almost a quarter of the patients achieved a complete response based on RECIST criteria. And initially, the first 24 weeks, our side effect profile was okay. It wasn't great. One of the big things that we noticed, and this was not in the TGCT patients, but in the solid tumor patients who were getting Pexi, we did have a patient pass away from hepatotoxicity, and we had one patient had a liver transplant due to that. But again, these were not TGCT patients. These were patients being treated for other tumors. But we did note that there were continually elevated aminotransferases in a lot of these patients. And so that was a big concern, especially with the first 24 weeks of treatment. As we went into the longer term treatment, what we found was the hepatic abnormalities actually occurred in 95% of patients, but 92% just had the elevated liver function tests. And then there were 2 hepatotoxicity is the second type, and there was a 3% rate of that. All of those ended up being reversible, but it could take up to several months for the enzymes to normalize. Within the study and that within then the 39 months, the long-term data, we did notice that 68% had a dose reduction or interruption, but this was all comers. So people who withdrew consent or people who had disease progression or were non-compliant with the treatment. Only about a quarter of people had to either DC the treatment or dose reduce it due to adverse events. So the problem that we're sort of getting to now with using Pexi is that we have the FDA indications, but should we go further with this? This is a patient of mine who came in, 66-year-old female who'd had multiple surgeries on a TGCT of her foot before she came to me, and she came to me March of 2020, and this upper right hand MRI was her disease at the time. We put her on Pexi, and then in July, this is the right lower corner MRI, and so she had no detectable disease at that point. She's currently on a much smaller dose of Pexi. She's on 200 milligrams of BID rather than the 800 that we were doing in the Enliven study. And I honestly do not know her liver enzymes, but she's had very minimal symptoms or side effects from the drug itself. So our unanswered questions, patient selection, should we really just be doing this on unresectable diffuse type and recurrences, or should we consider this in initial patients that we have a concern that they might recur? How long? As obviously the effects seem to be better and better the longer the treatment has gone on in these first 39 months median. But when would we actually stop for some of these patients, and would it be different for different patients and different responses? And then learning from the denosumab studies, what will we see long-term? And will we eventually be using this as a neoadjuvant drug, and will it make surgery a little more complex in the future? And finally, the last question is, who should really have ownership of this? In orthopedics, we see the full spectrum of disease from nodular type to diffuse type TGCT. Medical oncologists really only see the diffuse type, but they're probably more equipped to deal with things like the liver toxicities. So those are some of the discussion points and things that we still have to figure out from this drug. And that may have been quicker than 8 minutes, but hopefully that'll help catch you up a little.
Great job, Brooke. Appreciate it. Obviously very exciting to have something for these patients that we've struggled with for a long time. I think Dr. Schwab was unable to make it to give a live presentation, but does have a recorded presentation. Dr. Schwab, if you're there. There we go. I think we're just gonna push play. He's gonna tell us about immune checkpoint regulators, failures, successes, and side effects. I think we need some volume. Yeah, we can't hear it. Yeah.
I can't hear anything. Can you hear anything?
No.
No.
So
He looks good, but
yeah,
I was hoping for more.
we should also hear something.
Too relaxed.
Yeah, look at how calm he is.
Agree.
Molecules are very important with regard to the functioning of the normal immune system.
Great. Can you start it over
Checkpoint
and turn it up even more?
in the pipeline. My name is Joe Schwab, and I've been asked to speak about immune checkpoint regulators, failures, successes, and side effects. My name is Joe Schwab, and I've been asked to speak about immune. When we talk about checkpoint inhibition, it's important to remember that we're not just talking about PD-L1 and CTLA-4. There are lots of checkpoint inhibitors, and there are lots of drugs being developed against multiple checkpoint inhibitors, particularly in musculoskeletal tumors. B7H3, which is an antigen that I study, is highly expressed in many musculoskeletal tumors. So it's important to understand that there are many of these drugs available, and there are many more drugs in the pipeline. It's also very important to understand that although we think of these checkpoint inhibitors in the treatment of cancer, of course, checkpoint molecules are very important with regard to the functioning of the normal immune system. And so when you give a patient a checkpoint inhibitor, you are releasing the immune system to attack the tumor, but you're also potentially releasing the immune system to attack the host's normal cells. And that can lead to side effects, which of course I will talk about. As I said, the checkpoint inhibitors have been extremely successful in select patients, typically in patients in carcinoma who express checkpoint inhibitors. Who express the checkpoints. But you can see that the success has not gone unnoticed. There are lots of drugs being developed. These are just some of the drugs that have been FDA approved, and there are, of course, many more coming. In order for a checkpoint molecule to work, there are several requirements. You have to have a tumor-specific antigen, and then importantly, you have to have a functioning HLA class 1 antigen complex. That complex is expressed by cells, and that expresses the antigen to the immune system, specifically to T cells. The T cells then have to recognize the antigen, and then importantly, the T cells have to be activated, and this is where the checkpoints come in. If the T cells are suppressed at this point, this is where tumor cells can continue to grow. Checkpoint inhibitors act at this level. But it's not just that individual cell, you're also trying to get the immune cascade to be activated. And this again is where T cells are activating their neighboring T cells. As I said, there have been some very profound successes particularly in carcinoma and in select patients. And that has not really been true in sarcoma. There have been some successes, but there's been lots of failures. If you look at the initial trials, which focus primarily on CTLA-4, PD-1, and PD-L1, the results were relatively poor. The initial studies in CTLA-4 found really no responses. And then when anti-PD-1 therapy, anti-PD-L1 therapy started to become in vogue, Still, response rates were relatively low. They did see some responses, in fact, 2 complete responses in undifferentiated pleomorphic sarcomas and in some dedifferentiated liposarcomas. But in general, the response rates remained relatively low. A bright spot had been the alveolar soft part sarcoma. There have been some nice responses, up to 50% in small series. When you look at a large group, it hasn't borne out to be. So the next step up, as opposed to monotherapy, is combinatorial therapy. Combinatorial therapy tends to work better, but the toxicities go up. And so if you look at the results here for combinatorial therapy, the results really haven't gone up by that much. Again, one of the hallmarks, or one of the bright spots, is alveolar soft part sarcoma. But even that, we're talking about relatively small numbers. And so in general, the sarcomas tend not to have as good a result as in a carcinoma. And that may be because we just have small numbers and we're sort of lumping all of these tumors together, as opposed to really pulling out the tumors that are most likely to respond. So what else could be wrong? Well, again, one of the fundamental issues is related to the decreased immune response to the cancer. So it could be that the original response is robust, but then the tumors escape surveillance. And one of the ways that happens is by decreased HLA class 1 antigen expression. As I mentioned before, that's a crucial part for these checkpoint inhibitors to work. You have to have a functioning HLA class 1 antigen system. And we talked before about this. I showed you that you had to have this HLA class 1 antigen presenting its antigens to the T cells. But what I didn't talk about was, what does it mean? What does it mean to express HLA class 1 antigens? Well, there's a whole processing machinery that has to be intact for this to work. And often it is the case that parts of this machinery are not working, or they're not expressed. It's typically not a mutation. The exception to that is in beta-2 microglobulin. There are cases where there is a mutation and the protein is just not expressed because of the mutation. But in most cases, it looks like it's related to epigenetic mechanisms, such as methylation of the DNA. You get decreased expression of the genes that are important for the expression of these proteins. And so you have decreased HLA class 1 expression. The nice part about that is that it may be possible, and it has been shown to be possible, to increase the expression of HLA class 1 antigens with various mechanisms, including things like HDAC inhibitors. So what are the bright spots? And again, the bright spots are not super bright, with the caveat that if one of your patients, or if you happen to be a patient that is one of these responders, then you're very grateful. So alveolar soft part sarcoma, when you look at all comers, it's about 1 out of 5, at least in the numbers that are available, about 20%. Same with vascular sarcomas. Again, I've sort of lumped these together, which is something that we do in musculoskeletal tumors, unfortunately. UPS, about 20% also respond. And then there's some success in dedifferentiated liposarcoma. There is a signal in some other sarcomas, which I will talk about. We are particularly interested in HLA class 1 expression, and I think it's very important to understand that in many tumors, HLA is just not being expressed. And you really won't have any success giving a patient a checkpoint inhibitor if they're not expressing HLA class 1 antigens. Chordoma is an example of this. We looked at 24 chordomas, and in 21 of the 24, there were components of the HLA class 1 antigen system that were not being expressed. And so none of those patients are going to respond to checkpoint inhibitors. And so it may be, and we don't know the answer to this, if you increase the expression of HLA, maybe they then would respond to the checkpoint inhibitors. That's an important point. Similarly, when you look at chondrosarcoma, which we did, we found some interesting results with regard to checkpoint inhibitors, but also with regard to HLA class 1 antigen expression. So paradoxically, the dedifferentiated chondrosarcomas actually have a very low rate of defects in HLA class 1 antigens. Most of the dedifferentiated chondrosarcomas expressed HLA class 1 antigens, as opposed to conventional chondrosarcoma, where 77 to 92% of the cases are missing components of HLA class 1 antigens. And this also correlated with T cell expression. So 90% of our D-diff chondrosarcomas have CD8 positive T cells, a robust amount of CD8 positive T cells, whereas only 20% of our conventional chondrosarcomas have that. And again, that correlates with HLA class 1 expression. And looking specifically at checkpoint inhibitors, a third of our dedifferentiated chondrosarcomas express PD-L1. None of our conventional chondrosarcomas express PD-L1. And looking again at B7H3, and there are drugs in development to target B7H3. Nearly 100% of our dedifferentiated cases have B7H3, and 70% of the conventional have B7H3. So there are other checkpoint inhibitors out there. But again, if you give this B7H3 molecule or inhibitor to conventional chondrosarcoma, it probably is not going to work because they don't have HLA class 1 antigens. So what about side effects? Again, we talked about the fact that you're releasing the immune system to potentially attack the normal cells. And one of the things that many patients experience early on is dermatitis. After the second dose, one of the major complaints, at least with PD-L1, is colitis. And this can be profound. Not a trivial amount of colitis, and it's a huge impact on these patients' quality of life. So these side effects do affect the patient's quality of life. From an orthopedic standpoint, arthritis is also a common complaint. It's not just their quality of life, these can be severe. So particularly when you start looking at combinatorial therapy, more than half the patients have severe side effects. And the rate of death associated with these drugs is about 1%. So it's not a trivial, these are not trivial drugs by any stretch of the imagination. In terms of toxicity management, usually it's steroids. So you're balancing this with giving steroids and suppressing the immune response. But steroids is one way to treat these with marginal benefit. If that's not working, then you have to stop the medication. So there really aren't a lot we can do other than stopping medication or steroids. To give you an idea of how severe and how real the toxicity is, we actually have an entire immunotherapy toxicity service. Carrie Reynolds, who provided some of these slides for me, is the head of that for MGH. And so I was actually not aware of that there is an entire service dedicated to treating these patients and their side effects. So in conclusion, checkpoint inhibitors are an important part of cancer care. We're all familiar with them. Their usage will continue and likely increase. They've been less successful in musculoskeletal tumors. Targeting patients with known checkpoint expression will likely help. Also, targeting patients with HLA class 1 antigens will likely help. Adverse events are common and they can be severe. Thank you very much.
Thanks to all the speakers for your excellent talks. I'm going to just summarize some of the comments and questions that have been posted so we can catch up on time. Kurt Weiss mentioned that the Department of Defense has a rare disease research program that does offer some smaller grants. So shouting out to all the MSTS researchers. Adam Levine mentioned also that the FDA has an Office of Orphan Product Development specifically for orphan cancer diseases that support clinical and translational grants. So again, bringing notice to those. There were two questions which have already kind of been touched on. Again, we have this open question as to how long do we treat patients with denosumab and Pexi, dosing, etc. And I think Brooke again raised the question of, are we the ones doing this, or should it be the medical oncologist, or of course, perhaps together? Dr. Iwata mentioned that the denosumab trial in Japan was terminated because of poor accrual. And Dr. Healy mentioned again that treatment with Pexi cost is a major impediment. If gonna take maybe perhaps one additional question from the audience, otherwise we're gonna move on to the next section.
I'll ask one question on Pexi, if you don't mind. Is there a thought that there'd be any benefit from zeroing disease, and then for these chronically recurring sort of knee, PVNSs, and then put someone on it to see if it would decrease that?
Can you clarify that? I guess, what do you mean zero?
In other words, if somebody presents to you, they've had someone in the communities, resected them 2 or 3 times. They've got a bunch of extra-articular and intra-articular disease that's recurred. They're young, they don't have arthritis. You just put them on the drug, or do you try and zero their disease and then put them on the drug to prevent recurrence? Is there any information on that?
I don't know that there's any data on it yet, but I think, that patient I showed in my talk, she had been multiply operated, showed up with recurrent disease. This was the 3rd or 4th recurrence for her. And so I started her on Pexi. I didn't think I really had much of a surgical option, except, we're looking toward like a ray amputation on that foot. So I think when you're getting to that multiply recurrent piece and your operations have failed a few times, I think it's worthwhile to start with the systemic disease, at least right now with what we know.
Great, thanks.
Thank you. And so we're now going to turn it over to Doctors Lin and Rose.
Well, good morning, everybody. It's been a fantastic meeting so far and program. It's my great honor, together with Pete Rose, to introduce our next session and speakers. We'll be talking about achieving equilibrium, a very important topic for all of us. And our first speaker will be Dr. Michelle Gert, who's professor at McMaster University, and she will be talking about the best and worst thing about my job. Michelle.
Thank you. Can everybody see the presentation? Can you hear me?
Yeah. Good. Yeah.
Okay, good, great. Okay, thank you. Okay, well, thank you to Pietro and the organizing committee for asking me to talk about what are the best and worst parts of my job. And so I put this talk together. I like to do my talks early, so I put this talk together in early 2020 before our anticipated specialty day in San Diego. And when I was asked to give it this year, I was very excited, and I thought I'd go through it and see if there's anything different. Did anything change in the past year that would make my job, the things I love about it and the things I hate about it, any different? And really it was just about the same. So these are my disclosures. I don't have any conflicts with this specific presentation. Okay, so again, I will go back to early 2020, and I really struggled and sat and thought about my years at Stanford and my alma mater and the statue I used to ride by every day to class, and how he just sat and thought and came up with ideas. And I had to really struggle to try to figure out what really is the best and worst part of my job. Is there really one best and worst? And there really isn't, because our jobs are so complex and so interesting that there really I couldn't narrow it down to one. And the other thing is that why am I different from anybody else on this call or in our society? We're all cut from the same stone, and we all love the same things and hate the same things. So whatever I'm going to say, is not going to be that new for you, but at least I do believe that we'll be able to, it will resonate with you. So instead of one, I thought, and last year was supposed to be an Olympic year, and perhaps this year will be an Olympic year. But either way, I thought instead of just one best and worst things that I love about my job, I'd do the top three, just to honor the Olympics and the bronze, silver, and gold medals at the Olympics. Okay, so we'll start off with the worst. The bronze medal for worst, and this I wrote over a year ago, is a gradual decline of financial support in my department for faculty focused on research. So I started my practice at McMaster in 2005, and I had a really strong department with respect to support in research and financial support in particular. And this sort of started falling apart about 5 or 6 years ago with government cuts to universities, and it trickled down to our department. And basically it got to the point where I was doing my research for free, and it's about half of my time. So it was very stressful for myself and my family. This was a very negative source of stress. And I know that many of us feel the same, that when our departments don't support us in what we want to do outside of clinical work. The silver lining is that there was a leadership change booked for July 1st, and that actually was delayed about 6 months due to the pandemic. However, that there is a new chair of my department of surgery. We don't have an department of orthopedics, we have a division. So the chair of the department of surgery has all the purse strings. And it actually turned out to be Mo Banderi, who most of you have heard of, who's super supportive of research. And basically I'm feeling the vice grip sort of release its grip, and things are going to improve. But that was a source of significant stress, and something I absolutely hated about my job. Okay, silver, we can all relate to this. When patients and families take their anger out on me at diagnosis, when it's actually your fault that they have cancer, and telling them that they have cancer and that you have a treatment plan is actually, you're the closest person to get very angry at. But what I've found with these patients, that if you're just patient and you give them time, they end up actually doing very well and can have very positive outcomes. And some of them end up being my favorite patients. So for example, one patient I recently had, I had to get risk management in to see the patient with me because she was so angry about everything about lining up in front of the hospital for the COVID screening, everything, angry. And everything went very well with her treatment. And she recently brought me a coffee maker as a present. So there are ways you just need to be very patient, but that definitely makes you want to quit when the patients blame you that they have cancer. Okay, the very worst. When young patients die, when children pass away after we've treated them, these are some cards that I got from families or patients before they died or after their children died, and thanking me for their care. But definitely, there isn't anything worse than failing in your treatment with a child and they pass away. So I think anybody who works in oncology, pediatric oncologists are sort of, they're inured to these outcomes, but in orthopedic surgery, we're always looking for the win. So when we fail in these cases, it's horrific. So that is the worst. So I do have a little bit of a bonus, a little bit more lighthearted. When we get urgent referrals for lipoma and MRI like this, and suddenly it's an emergency, and I have to see the patient right away and take it out right away. So this is just presses my buttons that you know, a radiologist cannot make simple calls on a lipoma. So that's not a terrible thing. It's just annoying. Okay, the best. Let's be a little more positive. Okay, bronze. I know everyone here can relate to this. Every day is a new challenge. It is so interesting. My mantra is it's never boring. It's never boring in orthopedic oncology. There's always something going on. So we have to figure out how to resect tumors successfully, save the limb, save the life, save function. It's every single case is different. It's fascinating. And each patient is complex and is managed by a group and a team, which is, I find, very interesting. There are some diagnostic dilemmas, trying to figure out, you know, you're a sleuth, you look at the x-ray, look at the patient, you try to figure out what's going on. It can be really fun. And of course, writing and reviewing research grants sounds like it's not fun, but it actually can be, because it's very challenging, and these curve to improvement is quite enjoyable. Same thing with writing and reviewing papers. It's a lifelong learning experience, and it's something that, you know, as you get older, we find our bodies are no longer as good as they used to be, but our minds can be better. And that's why I really like to grow in my profession. And I do really, really enjoy working with trainees. Okay, number 2, operating. I know everyone on this call would agree that surgery is the most alluring profession. You never feel so alive as when you're operating, and it is an absolute pleasure. And I find that every day that I operate, I feel very lucky. I'm so fortunate to have the opportunity and the right to operate, because it is just something that we've all been probably obsessed with since we were kids, and to actually be able to achieve that ability to operate and be surgeon is something that I'm very grateful every day. And the best are the teams I work with. There's no question that the people that in your life and in your work make your job. So this is my clinical team at a Christmas party before the pandemic. So this was the Christmas party between 2019 2020 with my kids and my research group and our clinical work, and my partner David Wilson, his wife and his kids, and we just had a great time. So this is my clinical team. This is the core research team at McMaster. We're having some champagne in the hospital after University of Toronto randomized the last patient in parity. This was in October of 2019. And the team has grown a little bit, and there's some more people now that are working on the safety trial with us, but this was the core research group at the time. And some of you will see yourself in these pictures. The teams I work with expand internationally. We've gotten together all over the world and had a wonderful time together, and really touched each other in ways, in personal ways, talked about our families, talked about our lives, talked about our practices, and really made connections that will last basically your entire lifetime. And so this is in Athens, the group. On the left, this is our colleagues in Singapore. On the right was the ICM Philly Orthopedic Oncology team with representatives truly from all around the globe. who we work together to hash out the recommendations for infection prevention and treatment. This is the group in India, a very powerhouse research group, and our colleagues in Sao Paulo, Brazil. And then we met up in Hungary on the left. These are my dear friends, Eduardo and Roberto in Spain. And this is my favorite picture. This is in l'Abbaye in France. And you can't find a single person in this picture that's just not having the time of their life. It might have had to do with with the French wine and the French food, but we were just having a wonderful time. This picture and all the pictures I've just shown you have a much more poignant purpose now that we are not able to do this at the moment and have not been able to for a year. And the bonus, the best, are the grateful patients. I'm guilty of absolutely loving getting cards and presents from patients, not because I need the presents or I need the cards. It's because they're showing me that they are grateful and that the work that we do makes a difference in their lives. Thank you very much.
Michelle, thank you very much. Our next speaker is going to be Parker Gibbs. Parker is a professor of orthopedic surgery, as we all know, at the University of Florida. He's actually the Eugene F. Jewett Professor, the Chief Medical Officer for UF Health Shands, and also the Chief of Orthopedic Oncology there. So Parker, thank you very much. You're going to share with us a talk on balancing research and clinical care, which obviously ties into some of the themes that Michelle raised as well.
Thanks so much, and to the rest of the organizers for inviting me to chat and officially labeling me as old, given that I have to give this perspective talk now instead of actual data. So thank you. Real quick. Oh, maybe not. There we go. No real disclosures of significance. The talk is about balance. And this is a little idyllic view of balance, probably what we all thought when we were 8 or 9 years old. And I'm afraid I'm going to have to disprove this lovely picture. So why would we even try to do a career that balances both research and clinical care, right? Because we think it's fun. We actually get to engage people you might never meet otherwise, as Michelle just showed in multiple pictures. We get to meet people that are very, very different from us as surgeons, specifically orthopedic surgeons. And we embrace a challenge. We get to be creative, and we might actually be able to make a difference. But why not? Well, it's hard. It's really hard to do this research clinical care act. It's humbling. Oftentimes our regular orthopedic colleagues laugh at us. Surgery is a full-time job. Oh yeah, and research is a full-time job. And we have better things to do on our nights and weekends. When we think about balancing, there are lots of things to balance just besides your clinical care and your research time. There are many, many competing interests. Your patients, your partners, both in clinical activity and research activity, your RVU target for those of us in the United States, our families, our friends, and then even our residents and medical students, graduate students, and ourselves. So that's a whole lot of stuff to balance, and I'm only going to talk about 2 of them. When we talk about balancing research and clinical care, there's some math that doesn't make sense either. So if you are fortunate enough to be a clinician scientist and you say you get half your time to do research, so you're a half FTE in clinical care and a half FTE in research, that adds up to about 1.5 full-time equivalents of time. Conversely, if you have a half FTE of research and a half FTE of clinical care, that adds up to about 3/4 of the productivity you might enjoy if you were 100% in either. So the odds are a little bit stacked against you in time spent and productivity produced. And if you're going to do this, you need some help. You actually need a lot of help. You need a good chairperson. You need a good clinical partner and or mentor. And you certainly need a research partner and or mentor. And all these people need to be relatively selfless in their support of your mission. I've been very fortunate. I've had 2 chairs since I've been at the University of Florida. Peter Guerin and Mark Scarborough, who's going to talk later. It's really embarrassing to say good things about Mark, but I will. He's been incredibly supportive. And what chairs provide you is time. They give you a little bit of reality of what's expected of you in clinical productivity. They need to believe in the mission, as Michelle mentioned just a few minutes ago. And unfortunately, they have to give you a reality check on salary. So if people expect to spend some time in research, it doesn't remunerate quite as well doing clinical care. And so that's a harsh reality most of us have to experience and appreciate. I'm fortunate that I have 3 clinical partners pictured here. All of them believe in the mission. All of them have my back, and I've got theirs. And it really takes that communication and that support for both missions and all of the partners in your division to make sure that that kind of research mission goes forward. Research partners, these are folks in different times of my career who've been my partner in research, and they have the same qualities. Dennis Steinler up in that upper right was my original, he's a neuroscientist, believe it or not. Steve Givzani in the middle, kind of a mentor of mine in the middle of my career. And then Greg Sawyer on the bottom right, who's a friction physicist now. And then the guy on the bottom left, who's just been my sidekick for the last, I don't know, 12 to 13 years. But all these people, again, they understand where you are when you're not in the lab, and they've got your back. So if you look at your clinical life, your patients come first, always, and your partners comes next. I'm going to get on a little bit of soapbox about being honest about your clinical skills, because in my role as CMO here, occasionally I see clinician scientists who are fresh out of fellowship and want to do a lot of research time and think that the clinical part of their world is fairly straightforward. It's not, especially in our careers. I think the first 5 years of your clinical practice are critically important, and you need to be available to hone those skills so that you can later really spend some time in the lab. But be very brutally honest about your clinical skills and get help when you need them. The other thing you have to remember is that your life is one big favor ask, and all those people in the lab and all those people in the clinics and the ORs are doing you a favor, covering for you while you're doing other things. So make sure you communicate well to all of them and often. Just a couple pearls: beware the half day. So when you have a half day clinic, you know, that starts at 7:30 in the morning, supposed to end at noon, it never does. And so you end up losing 3/4 of the day. And so I try not to do that. I try to do whole day segments if at all possible. Beware the urgent case. That's why you have friends. The patient that shows up that needs urgent care and you're scheduled to do some big assay in the lab. Beware the take back, the patient of yours that has a complication that you have to take back to the laboratory, take back to the OR. Again, you need friends to help you with that if you can't do it yourself. Ask folks to help you field phone calls. Hope that you have partners that you can plug and play with, and inform your patients. This is really critical. At UF, we're very fortunate to say that to our patients, hey, if I can't take care of your emergency because I'm somewhere else, I have other partners that will. And as long as you inform them of that, they're almost always okay with it. And then remember that you can't do it all, but you're going to try. Advance that. And then your research life, remember to express gratitude, take time to read and to think, and structure your time. When are you most alert? Because in the lab, the stimulus comes from within, unlike the clinical world where it's external. And so you need to be there at the time when you are personally most alert. And then obviously you need to try to protect your time, be there when the real scientists are there, and don't be a surgeon jerk. And there's just a final uncomfortable truth. The folks against whom you're competing for dollars, they don't care about your work-life balance. They don't care about your clinical research balance. They wake up, eat, work, sleep, and dream science. They don't have to deal with a clinic or an OR, and they're your competition. So try to make some of them your friends. We lose efficiency moving from one world to the other. The worlds blend, but you have to restart your brain every time you go from the OR to the lab or the lab to the clinic. And that's where that loss of efficiency happens. You have to accept that your schedule won't be perfect, and you have to be present when you are there. So one thing in the lab, you want to be fully immersed in the lab and present when you're in the lab, and fully immersed and present when you're in the OR. And I violate that rule every single day. It's okay to feel like you're not giving enough to either arena. You're never where you're supposed to be. That sensation, you feel like you're playing hooky in everywhere you go because you're not giving everybody the effort they deserve, and that's okay. Both of these worlds have journal clubs, national meetings, social events, and so that's double the number of those that you might normally have to, and you're going to have to choose between the two, and that's okay too. And then it'll really hit you because you won't be able to teach residents as much. You may not be the go-to person for surgery all the time, and that's going to hurt you, and that's also okay. So I need to play that. Here's the teeter-totter again in real life. This is what balancing a clinical and research career is like. It's not quite that idyllic picture of two children in a little suburb. It's more like that. So is it worth it? Nope. Bottom line, just has to be fun. There are a lot of pearls. Happy to talk to anybody anytime. Hope I kept that under 8 minutes. Thanks so much for your attention.
Parker, thank you very much. Michelle, thank you as well for your talks. Pietro, Nick, and Carol, I think we're supposed to move on without discussion now. Is that correct, to stay on time with the schedule?
Yeah, there's no discussion for this session, and also we are 10 minutes late, so I would suggest to move to next part, next session. There's some new technologies, if you agree.
Unless you want to co-moderate it, because the co-moderator isn't here. I
Okay.
co-moderated for my other meeting.
Okay. Okay, I don't see my co-moderator, Dr. Carmody, so I'll ask Jay to co-moderate. He's right beside me here.
But as per Parker's talk, I'm in the OR and I have to go to administrative meetings, so I'm going to let Peter run the session.
And I'm in the OR and I got fellows doing it for me too. So, okay, so enough of that. We'll jump right into new technologies, innovative surgical techniques. And I'll start with Francis Lee, minimally invasive endopelvis reconstruction for metastatic cancers.
Hello, good morning. It's really great to see you and hear your voice. And today I'm going to share some fun experience in minimally invasive pelvic surgery. So no introduction, iliac crest.
Can you share your screen, Francis? Can you share your screen?
Oh, yes. I think I did. One more time. Can you see me now? Can you see presentation?
Your presentation.
No,
we can see your beautiful face, but not your beautiful talk.
Oh, I apologize. One more time. Here we go. What about now?
That's good.
It's better than my face then. That's great. So no introduction, iliac crest. Why iliac crest? You can feel it. It's the strongest bone, tricortical cancellous bone, and it can lead you to the acetabulum. And my lab has been doing some research how metastatic cancer cells affect bone homeostasis. They are really bad guys. They hijack bone homeostasis. They inhibit osteoblasts and increase osteoclastogenesis. As a result, systemic denosumab or Zometa do not work all the time. So my suggestion, we have to kill the cancers because they are the main culprit of osteolysis, and maybe we may apply Zometa locally. I did some Feynman element analysis. If we use the screws and cement through the iliac crest, that provides the strongest support compared to screw alone or cement alone. Is this true? So we did some biomechanical study, and it shows that, again, the cement and the screw construct give the strongest biomechanical support. And this is more evident for cyclic loading. Screw alone or cement alone cannot resist cyclic loading well. So what's the message? We are using a Trojan cannulate screw. We have to get there, not from outside in through an open surgery, but through the horse. So my surgery is called AORIF. It's an ablation to kill cancers so that we can stop cancer-induced bone loss. And also heat can cause thermal denervation to decrease pain. And I use a balloon osteoplasty like a kyphoplasty so that I can create space for initial cement deposition. And also it can decrease bleeding from tamponade effect. And also it helps to extrude the cancer through the second cannulate screws. Then I feed the defect through the screw with the Zometa loaded bone cement, just like our colleague explained this morning. And I stabilize with the cement and the screw construct. And also the heat from cement somehow seems to denervate and the pain seems to be much less. And we can do this simple procedure called AORIF for any locations through the iliac crest, either in supine position or in prone position. I'm going to share 9 cases today. This patient had a bladder cancer with a painful mat in the acetabulum. This is how it works. And these are the intro pictures. Even though patient is obese, you can feel the iliac crest very well. And just throw the guide wire in and put cannulate screws. And through the hole, you can do balloon osteoplasty, thermal ablation, and the injection of a cement with the bisphosphonate. Somehow it does not advance. So this is the intra pictures showing screw constructs. And you can see filling of a cement around the acetabulum. I can say this is not perfect, but actually it's not a bad deal, given the fact this is a percutaneous procedure. And this is the post-op, 2 weeks. Patient has some limp because of the muscle atrophy, sarcopenia, and the pain. But he did some hip exercise, and even he can run in the clinic without pain. My second case, patient has a metastatic breast cancer. She has a severe limp. Again, we did a very simple percutaneous procedure. Throw the guide wire in, put the screws halfway. Then you can do ablation, balloon osteoplasty, and the cement injection using a kyphoplasty cement gun. And this is only 4 weeks later, and she can actually ambulate very comfortably, very different from the pre-op. And interesting, I don't know whether it's from Zometa or from cancer ablation. Pre-op, there is no bone here, but as time goes by, there is a dramatic bone regeneration. I don't know whether this is from Zometa or cancer therapy or ablation, but it can happen. So sometimes patients really pays. My next case, multiple myeloma in the anterior column. And she has quite painful limbs. When the tumor is in the anterior acetabulum or anterior medial superior dome, you may need a supplemental screw that targets the superior symphysis pubis. And she can ambulate very well through this minimally invasive procedure. My next case, 94-year-old female, unknown primary. I do not know her remaining life. And there's a high risk for anesthesia. So again, we did this minimally invasive airway procedure. She was not able to ambulate, of course. This is 2 weeks later. She's holding cane upside down. And this is a COVID time. That means the patient can go home the same day without staying in the hospital. And this surgery was actually very useful during this COVID pandemic era. Next patient, patient with de novo breast cancer with a widespread bone mets. What should I do? Should I start chemo first or salvage the hip with a megaprosthesis? We can even see the femoral head through the inner pelvis. And we did again minimally invasive superior medial column screw through the side of the pelvis and the conventional iliac crest screw so that we can build the entire pelvic construct as one unit. How bad is the cement extravasation? Well, she may have some vagation or some cartilage, but this is what she does. She does not really complain of any pain. I mean, it's very painful to get this CAT scan while there's some extravasation. But again, she was not she was able to have a chemotherapy without delay by not having big surgeries. Case number 7. How bad is the protrusion stapulae? Should we do it or not? The patient has a lung cancer and every day is important. And every single targeted therapy wants to kill even single cell disease. And this is an immediate post-op, but you can see acetabular incongruency and some proximal migration of the femoral head. And this is him. CAT scan shows like a leg prosthesis disease or advanced avascular necrosis. Well, that's what I'm. But this is a kind of aspherical congruency like leg prosthesis disease. And his function is pretty good. I mean, it's not bad. I call this as a semi-Gödel stone or a pseudo-chakra joint. Probably the heat and ablation may denervate some nerve endings. Case number 8. What would you do? This patient has new breast cancer with the bone mets. New. Should we start chemo first, or patient can be bedridden and can have inpatient chemo for 6 months or even a year? There is no bone. What implant can we use? As a bonus, if a patient has a femoral neck fracture. And we did the ARIF again, 3 days, 2 months, and 11 months later. There is a dramatic bone regeneration, even though in the beginning, the bone regeneration was not remarkable. And this is the plain x-rays. Terrible fracture. This is a kind of substandard care, but I think it's holding very well. And it's 11-month follow-up 3 weeks ago. She can ambulate very well. I'm offering her bipolar or total hip, but she says she'd rather have a continued oncology care to prolong her life. And if it really breaks, she may consider additional surgery. My last case. These days, we see increasing number of periprosthetic metastases. I call this as a pathological implant loosening. And this is how it works. And he also has a kyphoscoliosis. Should I do revision using megaprosthesis and everything in this 70-80-year-old male? And you can see actually bone is seen through the, again, inner pelvis. So in this particular case, I did percutaneous implant loosening revision. I usually use only 2 screws, but I throw in 4 screws to go both superior medial and the posterior wall. You can see a huge space that is represented by a balloon, and we injected the bone cement. Patient is from far away, and the patient was staying in the hotel next to the hospital. And he was able to walk quite comfortably, even though he has a severe kyphoscoliosis, without much pain.
Turn around.
So today I showed some 9 cases, and I have more examples, and I have a detailed information on their pain score, functional outcomes, transfusion, and all others. But I would be happy to present this in the future meeting because of lack of time. And if there's a time, I would be happy to answer any questions. Thank you so much.
Thanks, Francis. That was incredibly impressive. There's lots of questions. I don't know that we'll get to all of them. So we'll go ahead with the second talk. David Joyce, Navigation and Musculoskeletal Oncology, an update.
I just need to be able to share my screen. Yep. See if we get this to work correctly. Is that showing up?
Yeah, look, can you put it to full screen?
Yeah, I think it's, let me share the other side. Is that better?
Yeah.
All right, so I was asked to give I'm David Joyce. I'm from Moffitt Cancer Center. I was asked to give the update on computer navigation and musculoskeletal oncology. I was kind enough that Odion and Doug let me do this. I appreciate the society allowing me for my ability to communicate and talk. I have no conflicts. The only conflict that exists is we typically use Stryker and Doug is a Stryker consultant. So why computer navigation and musculoskeletal oncology? For the most part, some of it was developed based off of the fact that it's hard to make a determination about where bone margins are actually at. Looking at some of the studies, even experimental, there's only like a 52% probability of being able to get a 1 centimeter margin in 3 planes. And even in the past, on some of the previous studies, people recommended at least a 2 centimeter margin due to the inability to accurately identify where a tumor is at in the bone. Local recurrence in the past, at least in the pelvis, in a marginal resection has been up to 70%, and sometimes even up to almost to 92 to 100% in some other studies. Our adventure in computer navigation kind of started here at Moffitt Cancer Center and looking at what our early experience was, just as kind of a summary. We thought it could improve our accuracy of resections. We thought it improved the accuracy of reconstruction. And so where do we start? This is kind of the beginning. This is one of the first slides kind of presented. It shows you kind of, how a suite actually looks. A lot of this has changed over the future. And so we start off with reviewing some of our original literature that we thought helped us. So one, navigated distal extremity surgery or just extremity surgery was very helpful. We were able to pre-plan. Traditionally, we had to measure out with a ruler in the past, and now we can measure out with the computer navigational gun with preoperative imaging, make our determined resection as well as plan for our determined reconstruction length. It also allows us to appropriately plan for rotation. In a navigated distal femur, we can plan for the appropriate implant, which in the bottom right-hand part of the screen, you can see kind of an implant that's superimposed, but that's sort of how we plan for where our resection will be at. Precisely, we can measure from different points. It allows us to appropriately and change our margins based off of where we put our planes. And this is advanced over time to the point where obviously it's easy to get a rotation. Typically, we'd guess at our rotation in the OR when we're doing a distal femoral placement, and now we can actually plan it preoperatively and then measure it out and have it match. These are kind of two different images, probably about 10 years apart, of the way we actually ultimately determine our rotation. In that study, looking at distal femur, as we looked at leg lengths, ultimately found that basically all had good margins and then no local recurrences and improved accuracy of the reconstruction. And ultimately, the leg length discrepancy was pretty minimal, about under a centimeter. So we could at least establish where we're at. Where did we start with this? Originally, looking at the left side of the screen, this is what we started with as far as imaging. This is kind of a fluoroscopy unit, creates about 256 images that we then reconstruct into a CT of the pelvis or a CT of whatever bone we're looking at. And then now we've moved on to an actual intraoperative CT, which helps out immensely. And so we wanted to incorporate that into most of our hemipelvectomy. Previously, we would plan on sawbones, and now we ultimately plan on the computer where our resections are at. This is kind of how we started. And then with Doug here planning his resections in the past, using some of the same instruments. These are the instruments typically we use in the past. I've changed some of the instrumentation, which I'll update today to make things a little bit easier and a little bit more accurate. But we're able to basically plan, and that's probably the biggest benefit to computer navigation. This, for instance, is a navigated sol that they used to use in the past. I currently have changed some of that due to some of the inaccuracies you have with some sols, as well as control issues. We could easily resect these out and even restore leg lengths pretty close to where we wanted to start based off of the computer navigation. Lately, there's been pretty good data in the last maybe 3 and 4 years, especially coming out of the UK, where they've looked at computer navigation and does it make things safer for patients. With the navigated procedures, they had no intralesional procedures performed. With a non-navigated, they had an intralesional procedure about a quarter of the time. So the next question is the role in 3D intraoperative navigation. The duration of surgery was not increased according to some studies. Some studies would argue that the beginning obviously increases some of the surgery, but overall, everybody had a negative margin. Our data that we kind of reviewed here, which is kind of supported by the next paper that recently came out in 2021, is that it seems that we can oftentimes minimize some of our local recurrence rates, at least in the bone, compared to what the old recurrence rates were in the past. This is one of the most recent papers to come out this past year. They looked at 13 navigated, 37 non-navigated surgeries, basically found local recurrence rate about 23% for the navigated group versus 56% for the non-navigated group. And so that was clinically significant. They felt it confirmed improved local control and help for survival, hopefully in the future for these patients. So computer navigation resection. So what are some things that have changed and are going to be happening in the future? One of the benefits to the navigation is multiplanar osteotomy resection. So because of accuracy, you can start planning for new things, such as custom prosthesis. As we get better materials that can integrate into the bone, we can have more accurate resections to actually place an implant in there and not having to totally customize on the back table with an allograft. Just one of our surgeries putting in our new custom prosthesis in, and this is kind of what it looks like. In the future, most of these will probably be some form of custom prosthesis. The materials will all change, but 3D printing has definitely helped out as far as computer navigation. This is a 3D printed model, which for planning these, we kind of send them back and forth between the company, making where our surgical resections are, and have discussions about where the appropriate hardware should be placed as our screws pre-planning. This has probably decreased some of the time frame in reconstructing the pelvis. For those of us that reconstruct the pelvis, this is just a model with a component and reconstruction. So, you know, navigated surgery may be beneficial for pelvic bone resection and decreasing the risk of bone local recurrence. Some of the data sort of supports that now. May also improve reconstruction accuracy. We don't have the best data on that, but from anecdotal data from putting components in that are pre-planned, they seem to fit pretty well. And then it probably does not actually decrease the overall time of surgery, but that may change in the future. So what can we do also with navigation? Joint sparing resections are also another thing that we're looking at. So for tumors that are very hard to see intraoperatively, especially with fluoroscopy, can sometimes afford us the ability to change the type of implant or the reconstruction that may be made. This allows us to appropriately plan. Typically, I use a navigated burr now, somewhere around 1 millimeter to make the cuts a little bit finer. We can plan ahead of time, which also probably decreases our timing. And then we can appropriately mark our margins out that we want for our resection. We're able to place an allograft and reconstruct the patients and have that planned reconstruction fit fairly well and save somebody from getting a proximal tibia replacement. We can even do that when we're trying to avoid extensor mechanism failure or having to replace that, where we can appropriately plan and resect. So how do we get to where we're at? In the past, we've used saws in the past, and now we're using instrumentation that's a little bit finer. I currently use a burr to make it a little bit easier and a smaller osteotome. And one of the future tools that are probably coming up that we've kind of incorporated from spine, and I've used a couple of times, is an ultrasonic aspirator. Allows a little bit finer control instead of having the burr run on your hand or on the bone. Allows a little bit better safety, especially in areas that are precarious, such as the pelvis. And so what's the future state of computer navigation? Well, it's a good platform, allows to plan and export the possibility for a plan for engineers for custom components. Currently that doesn't exist, but that's where things can be heading. Finer resolution CTs and even MRIs, especially intraoperative, will allow for less error. Currently it's 1 millimeter cuts now, and I think they'll be decreasing to 0.5 millimeter in the future, which will help decrease some of the registration error. Improve instrumentation, allow more tools to be navigated. As orthopedic surgeons, notoriously, we're always borrowing from other surgery services to make things better for our patients. And I see kind of the integration of robotics with computer navigation to help us appropriately make resections, make them much cleaner, more efficient, much like they do in total knees, total hips, and even some of the spine instrumentation. So what's the appropriate bone margin? That's one of the questions I always ask myself, and I have more questions about navigation as I can continue to progress. And then how do we actually measure our margins? Should we have an appropriate standard when we discuss computer navigation margin resections? In conclusion, navigated surgery can be beneficial for many types of limb salvage surgery. It involves major bone resection, reconstruction, and joint sparing. Navigated surgery seems to improve the margins at the time of resection, and ultimately navigation may improve the accuracy of the reconstruction in the future. It's unlikely to help you with your soft tissue margins, although some people are looking at in different fields, such as liver oncology and kidney cancer, as far as computer navigation with soft tissue components. But currently in orthopedics, that's not anywhere on the horizon. And navigation is not the solution to margins, but is a tool, if used appropriately, can help us minimize compromised bone margins. Ultimately, I think as imaging gets better and we have a better interpretation of imaging that we can incorporate into the navigation, we'll appropriately choose our margins. And as components get better made and we have better bony integration, we'll be allowed to have our margins be further away in order to appropriately spare enough bone, but not get close enough to the tumor. Thank you very much. I appreciate you guys listening to me. And do we have questions yet?
Do we have time for questions at all?
I think we are we're running about 15 minutes behind, so maybe if as the moderator, if you have one question that jumps out at you, sorry to put you in that spot, but okay, maybe one question.
Problem. Thanks, Nick. Well, maybe I'll ask, there are a bunch of questions. Maybe I'll ask Francis and David to address them in the chat, but the question I have is for Francis. Francis, I mean, some of those cases you've achieved what us mere mortals could only dream of achieving. So there's obviously a steep learning curve. The question is, somebody starting out who wants to adapt something like this into their practice, what limits would you say you would put on this procedure in terms of what you can hope to achieve with the reconstruction?
I mean, our goal is really to avoid any complications from open surgery and get the best chemotherapy without delay. And I know there are some concerns about screw protrusion, and I try to stay short, like 1 centimeter from subchondral bone, just to answer Anna's question. And I think imaging is very important. I sometimes use a C-arm. I'm not a major guy at Yale. I just use whatever empty room. And I usually use a cardiovascular cone beam CT. But if you have a navigation system, I think that'd be a great bonus. But technique is very easy. I took all the pictures myself because my resident is putting guide wire, and they are very good from their trauma experience. And I'd be happy to share a PDF file or some movies with you. And just send me an email. I'd be happy to share everything I know.
Okay, thank you.
All right, thank you all for a great session. Dr. Sim, would you like to introduce the next session?
I'll be happy to do that, Nick. Thank you. I want to first congratulate the scientific committee in our society for this marvelous program today. It's been really fantastic. and welcome you to this last symposium, which is very interesting and enlightening. As you know, I've had the opportunity to witness the evolution of our, especially from the beginning. And there's one significant observation that's clear, is that each significant advance and strides have been associated with the development of our especially societies and groups. This has been shown to be true in every field of medicine. So rather than working as a vacuum as we did for the first decade or two, the cross-fertilization that's related to these societies that will be discussed by our panel has led to teamwork and the current standards we're enjoying in musculoskeletal oncology today. So we have a wonderful panel, and perhaps Nick, you could introduce the panel, and if we have time for questions.
So sounds great. It is my honor to introduce my dear friend, our Laura Randall, to give the first talk here on the role of MSTS. You're on mute, Laura, sorry.
There we go. Sorry about that, guys. My apologies. I had it up and then I closed it. So let me get going here. Can you see my screen okay?
I think you're sharing your whole desktop instead of just the talk, but that's fine if you want to do it that way.
Now you got it, right?
Yep.
Okay, so about that. So thanks everybody. I know we're short on time. These are my disclosures. None are relevant to this talk. There are some commercial vendors, but there's also some intellectual involvement. So obviously, MSTS is really a global organization, our partnership with multiple people. Through the Academy, we have formal educational forums through the instructional course lectures, case presentations, and technical skills under the amazing leadership of Val Lewis. And then more recently, Wakenda Tyler, we have really brought on a nice slate of talks. You can see them listed here. And I think the Academy and MSTS is internationally attended, and this provides a great forum for education. So sorry, guys. But we have a lot of things going on beyond our own MSTS annual meeting and the AAOS. There's ISOLs we'll hear about from Mark. There's MSOS. There's the Asian Pacific MSTS, the JOA, the Argentinian group, the home of the Rome of allograft, if you will. And we have combined meetings, and there's traveling fellowships and a whole lot more that we don't have time to get into. We just had the ISOLs in Greece and Athens. It was a fantastic time. I could hook up with some of my former fellows from around the world, which was really exciting. We had Professor Tachia and Kawai give their talks about the respective societies of the JOA and CTOS, and it was a really great forum. I was the ambassador for MSTS, and it was really wonderful. We look forward to the follow-up ISOLs in Inferior, California later this year. I'm sure it'll be a fantastic program down south. There is the upcoming, just this next month, the Asian Pacific MSTS meeting with my good friend and colleague, Dr. Ozaki, for this being held in Japan. This will be a fantastic forum. It is virtual, but I encourage everyone to be engaged. Japan has really been a thought leader, as we heard a little bit earlier from Dr. Tachia. It's really where I've learned a great deal about techniques that are done that we don't think about sometimes in the United States, such as the autoclaving, et cetera, et cetera. And then, of course, MSOS, we can see our program chair here, Piero, and many of his colleagues who we know dearly, who are thought leaders in the European Union that have really driven the field. We engage with them through the MSTS and MSOS combined forums over the years. And I would encourage everyone to try to keep an eye out on the MSOS programs, because there's a lot to learn from that group. And then, as I've already alluded to, the JOA, I know John Healy and Chappie Conrad and others have been ambassadors for years. I've gone over a few times and really enjoyed the interaction with colleagues from Japan. They are the passion engine of orthopedic oncology and do some of the things that the rest of the world sort of then takes and adapts to their own cultures and norms within their own societies. And they're just a fantastic group. And so for all the young audience out there, I strongly encourage you to look to the JOA for your educational enlightenment. And then, of course, as I alluded to before, down south, the Rome of allografting, if you will, the place that has the best allografting results globally is the Argentinian group, and they hold periodic meetings that invite ambassadors from MSTS and other, that you can see here, we had Dr. Palmarini from MSOS down there, and we had Lee Jays, and then Joe was down there with me representing the US. And it's just a really great opportunity to exchange ideas. And so we should all be looking to see what the Argentinians do in terms of orthopedic oncology. The combined meetings are really the standard bearer for our global coming together, if you will. We've had one with ISOLs in 2009, and then more recently in 2015 with a fantastic program by Mark and Parker that was really highlighting, and we usually get very august speakers to come and talk at these forums, and it's really quite terrific. And then, of course, CTOS. I was the president of CTOS when Dick Lackman was chair of MSTS. We had the combined meeting. It was the largest sarcoma-focused meeting ever back in 2011. And that's certainly potentially on the queue in the future. Dr. Kawai, a world-famous orthopedic oncologist from Japan, is also another orthopedic surgeon with Jay Wonder, and Mark Gephardt as past presidents of CTOS. And then just a shout out for traveling fellowships in general. This is the AOA fellowship. That's something called the ABC, American, British, Canadian. I've highlighted some of the ambassadors from that program here, but this is certainly not inclusive. And I'm not meaning to speak about the ABC, or this is not necessarily about the AOA, but traveling fellowships really are critical either domestically or internationally to gain insights on how things do. I remember back in 2005 when I did this, I went over to Limpopo in South Africa with my cDNA expression arrays, and we talked about genetically engineered mice, and I had compress and all of these techniques. And the audience in these sites were just glazed over and had no ability to relate. And I was obviously tone deaf. And what I saw when I went over there was all of the rotation plasties, and I saw a technique I hadn't really had much experience with, claviculoprohumeri. I saw these kids who are orphans of the state running around playing basketball on the rotation plasties or the rhizomelic upper extremity from their claviculoprohumeri, technologies that were afforded only by plates and simple orthopedic implants, but biologic reconstructions were quite durable. And it's been career changing for me. And the only reason I had that, it was through global engagement in the realm of traveling fellowships. So with that in mind, I just want to close out by saying the MSTS fellowships are really getting an overhaul thanks to the executive committee now with President Carol Morris. But there's been many people who have been behind this process for really gaining a more intimate control, if you will, of the quality of our fellowships. There's going to be an MSTS sponsored match, no longer the SF match with Hip and Knee Society. And there's going to be an accreditation process to really make sure we have quality education for our fellows. And I'm going to close out by quoting our past two education committee chairs, because their vision on behalf of MSTS that have really moved us forward. And Dr. Val Lewis has been championing this exchange program for the fellowships that is emerging, and I'll quote her: "This exchange will provide an excellent opportunity for MSK fellows in training to gain additional education and oncologic perspective, expand their personal and professional network, new mentorship opportunity, and prepare the future challenges in leading and improving the specialty." Now that certainly is a domestic program, but who knows, as we grow, maybe we could become international as well. And then Wakenda, who is our current education chair, this is a long but very thoughtful quote. I'll just let you all read it, but it just shows that the MSTS commitment to 2, education domestically and internationally is first and foremost on our agenda. We have rich relationships with other organizations, and we really are one community, and we need to be working together. And with that, I'll close out and just invite all of you to Superior, California at your leisure. All of these sites are all within an hour or two, or maybe Yosemite's 3 hours away from SAC, but this would be a great place to host a meeting. We've got Tahoe, Napa, and the coast all in our backyard. So with that, I will stop and turn it over to our session chairs. Thank you. Sorry for the slow start.
Or for a great overview, and we're going to move from the MSTS to talking about MSOS. So Andreas Leitner, if you don't mind taking us to the MSOS perspective, please. I think, Laura, if you can stop your Zoom screen share. Yep, perfect. Thank you.
Thank you, Mr. Chairman, ladies and gentlemen. Thank you for the kind invitation to talk here on education and the role of MSOS in Europe. This is my disclosure slide. Let's go back 35 years or more to the ENF Treaty. You can see here Ronald Reagan on the right side and Mikhail Gorbachev on the left side, and they're signing the Intermediate Range Nuclear Forces Treaty. And for the first time, the superpowers agreed to reduce their nuclear arsenals. I think this is one of the beginnings of the end of the Cold War. And this Cold War plays the topic in my talk. Because in the same year, 1987, in this nice 500-year-old Venetian villa, Renaissance villa, 19 sarcoma experts met and founded MSOS. And one of those was, of course, Mario Campanacci, one of the most prominent figures in European sarcoma history. And you can see here the beautiful library of the Rizzoli Institute in Bologna, Italy. But one of those guys also was from the Czech Republic. And at this time, there was still the Iron Curtain between the West and the Western countries and the Eastern countries, of course, including Russia. It was quite difficult to cross this border, but Professor Matyovsky was able to come and was able to also be a founding father of MSOS. And from the very beginning, this was quite important because the aims of the European Musculoskeletal Oncology Society are to advance the science and practice of the diagnosis and treatment of bone and soft tissue tumors, to promote basic and clinical research, and to disseminate knowledge in order to provide a common high standard of musculoskeletal oncology. And the particular purpose of the association is to promote mutual collaboration between different specialists and institutes involved in the treatment of musculoskeletal tumors. And the first meeting 1 year later was in the neutral zone between the West and the East. It was Vienna, organized by Professor Kotz, and it was the first MSOS meeting. We then had annual meetings, and even 3 combined with MSTS, 2 in the US, in Boston and Washington, and 1 in Bologna. But we also went to the East. We went to Moscow, we went to Warsaw, we went to Hungary 2 times, and we will go to Kiev in Ukraine. And this year, if you would like to come this December in Graz, again in the, let's say, neutral zone. We have at the moment over 200 members from 41 countries, mainly Europe, but of course some from US or Argentine. And if you look at the map here from Europe, you can see here there's a concentration in Italy, Switzerland, France, Germany, Austria, and of course Great Britain. But we also have 8 members from Ukraine, 2 from Moscow, and so on. So I think this is quite important. Not only orthopedic surgeons, but also pathologists and oncologists, but mainly orthopedic surgeons. Concerning education, Professor Miklos Shenbroy from Budapest introduced the basic instruction course in bone tumors in 2015. It was very successful, and this was taken over by Professor Ruggieri. The course went then to Venice and later on to Padua, and only COVID was able to stop this course. But Professor Ruggieri also organized a course in Sarajevo, in Bosnia, where there's really a need for good education concerning sarcoma treatment. We also have a traveling fellowship, and you can see here 1 to 2 fellows per year. And I think it's quite important because they have to visit 2 to 3 sites and then have to look how sarcoma treatment is done there. And also they should try to start a study. And I think combined studies are very important also for education, because then you have a common language how to treat, let's say, adamantinomas or angiosarcomas of bone. And these are all 5 publications from 2020 on expandable tumor prosthesis, on COVID-19, and also on bone sarcoma care in Europe. And these are the studies still going on. So if you're interested, please visit our website. There's quite interesting studies, and of course, most of them are retrospective in type, but nevertheless, I think they're good studies. From the very beginning, we've tried to include also nurses and our professions, as they are especially active in Great Britain and the Netherlands and the Western countries. And you can see here that this year is the 21st EMSOS and L Professions Group meeting combined with the EMSOS meeting. And what I started with the Cold War, and I will end with the Cold War, because you can see, or you will see, that there is still something to do. And this is a very interesting publication from Cancer Epidemiology 2018. And this is the age-standardized 5-year relative survival rate for bone and cartilage sarcomas. And you can still see there is an iron curtain between the Western countries, Northern Europe, Ireland, Central Europe, and Southern Europe, with survival rates going up to 60%. And this is Eastern Europe with about 40%. So there's a big difference in survival concerning bone and cartilage sarcoma. And you can see here the trend went down after the fall of the Eastern Bloc. It's going up again, but there's a big difference still to the Western countries. It's a little bit better for soft tissue sarcoma, but nevertheless, you can see also here the Eastern countries are lagging behind. So take-home messages from my side: we need conferences, we like them. We need instructional courses. Hopefully after COVID it's possible again. I think it's very important to involve nurses in our professions. The traveling fellowship introduced by Graham has really been proven successful, and I think we need to join publications. But I think most important, we need a special focus on Eastern Europe. Thank you very much.
Thank you. Beautiful talk, and the data is great to see and teach us where we need to focus. So thank you for that. If we can go back to Gainesville for our last speaker, Professor Scarborough, will you take us home?
All right, thanks, Nick. Can you hear me? And can you see the slides? The thumbs up? All right. So I've had the pleasure of being the president of ISOLS and hosting a meeting in 2015. And I've also been able to attend most of the meetings since for almost the last 30 years or so. So I'm going to give you my perspective on the intercontinental role of ISOLS. The purpose of ISOLS is really, you can read it yourself, but to promote limb salvage in a kind of a multidisciplinary fashion throughout the world. The history of ISOLS, it started in 1981 with a small meeting, 127 people in Rochester, or in at the Mayo Clinic, and they really focused at the first meeting on prosthetics, modular implants, porous coating, and really looking at limb salvage from that perspective. The second meeting 2 years later was hosted by Dr. Kotz in Vienna, and they again focused primarily on prosthetics. They were also credited with as the first group to really kind of get the the social aspects of ISOLS started and really bring the world together, and they set the bar quite high for entertainment and camaraderie. 1985, Dr. Inneking hosted the third meeting, and at this meeting, it became a much more multidisciplinary focus of ISOLS. Curiously, in that meeting, they started talking about the use of methyl methacrylate for giant cell tumor, and it was one of the first meetings where they even talked about the use of chemotherapy for osteosarcoma. But at that meeting, again, it really was, it kind of brought the multidisciplinary focus of ISOLS to the forefront. The structure of ISOLS, it's very multidisciplinary. It's open to surgeons, oncologists, radiation therapists, engineers, nurses, ancillary personnel, and anyone really involved in limb salvage surgery, as well as research and product development. The meeting is every other year, so it's not every year, it's every other year, and it alternates between North or South America, Asia or Australia, and Europe. The administration group is in Austria, and the board is multinational. ISOLS also, besides holding a meeting every other year, also sponsors an educational fellowship, a traveling fellowship. This is the map of where the programs have been over the years and the list since 1981. And the stars are the ones that I've been able to attend. So I've been to most of them since I started my career. And you can see they've been throughout the world. So why is international collaboration important? It really provides a great forum for exchange of ideas. ISOLS is very multidisciplinary, it's multicultural, it's an educational event, and it's really, I think, clearly resulted in improvements in limb salvage. But you can't discount the idea of the human interaction, the camaraderie, the networking, the friendship building, the kind of the bringing together of the world, which to me is perhaps the most valuable part of ISOLS. It also is a forum for providing standardization of nomenclature in a multilingual world. So we have all these things that we have to talk about and use for understanding our field. So staging systems, functional evaluation, radiographic evaluation, tumor grading, outcomes measures, promise measures, response to therapies, classifications, et cetera. And that it really provides a kind of a unified language in a very complex world. The international education component of ISOLS is also very important. This is myself and a couple of our visitors from the Philippines that I met through ISOLS, and then they came and spent time with us at the University of Florida in Gainesville. We've had visitors from all over the world to our program, and many of the people on the program today have also shared resources and provided a forum for international travel. And those connections are invaluable. It really makes a difference in how we think about our field. And without that, we would not be connected. The fellowship is important. There's a 3 to 12 month fellowship that's sponsored by ISOLS, allows somebody to do a traveling fellowship, 1 or 2 people at a time, provides a stipend. Applications can be obtained through the ISOLS website, and then they report on their experience at the ISOLS meeting. There's a direct relationship between industry and the surgeons and physicians and others associated with ISOLS. This has been since the beginning of ISOLS. The first meetings really focused on limb salvage and prosthetics. And it's been a continued theme, and I think it's been incredibly valuable to advancing our field. The bar has always been set really high for the cultural and social events at ISOLS, and I think this is not only are the meetings good, but the entertainment's been spectacular. Some of the highlights, and I'd be remiss if I didn't mention some of the really good ones. So the Vienna Opera House, Mont Saint-Michel in France, the Great Barrier Reef in Australia, Seoul, Beijing, Kanazawa. Personally, ours, Cirque du Soleil, I kind of liked, and the events in Athens were spectacular. But those events really are often where you get to meet the people of our field. So why ISOLS? ISOLS is rewarding. It's truly educational. It's inspirational, and it's also quite entertaining. The last slide is that there's the 2 upcoming meetings. Fran Hornacek is going to host the next meeting. He actually announced a couple hours ago that the meeting has been postponed, and it's going to be in May of 2022. The new date will be announced shortly. And then Peter Stedman, one of our friends and colleagues from Australia, is going to host the 2023 meeting in Brisbane, Australia. So that's it for my talk, and thank you very much.
Thank you very much, Dr. Scarborough. I think just we're running a bit late, and so for these talks, I think we will forego questions. These were very informative, and I just want to make the editorial comment to thank the MSTS board. I think the idea that you guys got together and decided that hearing from folks from MSOS and ISOLS speaks volumes of kind of breaking down the barriers between the organizations and figuring out ways to highlight the roles of each and how we work together, and rather than see each other as somewhat competitive. So thanks for putting this, approving this and putting it on. Let me turn it over to my friend Pietro Ruggieri to start with the concluding remarks.
Thank you, my friends. I think we have been exactly in time, or almost in time, 2 minutes late. And I personally, maybe I shouldn't say that, because I, together with David and Nick, we are part of the team that organized it, but I really enjoyed the day. I think it was great, but what made this great, we just did a frame, but the picture is what the excellent, prominent speakers were able to give us. And it was not only interesting, but it was also a pleasant afternoon. I really enjoyed it. And I personally thank all of you, my dear friends and speakers, that have been able to keep all of us awake, although these many virtual meetings are sometimes destroying or annoying us, but I don't think it was the case of this afternoon. Before giving the word to Nicholas for the closure, I would like to say one more word. I thank you all, but I have a special word of appreciation to my mentor, Franklin Sim, because as I always tell him, all the things that I've done good in my work, in my career, I owe to this man, and the bad things, I did them by myself. Okay, so Nick, please go ahead.
No, and I don't want to keep folks past the time, just to thank everyone. As you saw, the kind of theme here was a more holistic view, was the sort of attempt here, whether it's with the different organizations working together, with having kind of a balanced approach to the new science that's out there, to the new drugs and integrating pharmacology and kind of biologics into surgical care, and the new techniques that you know, Francis Lee and Dave Joyce took us through. I think it was, I really appreciate everyone's efforts in this. And I think it's just a concluding remark. I'm going to echo Dr. Ruggieri. For me, basically everything has been colored by the loss of Jeff Eckhart this year. And I just want to say I can think of no one who more valued this group. And you know, I spent a lot of time with him, especially in his final years, and he loved everyone in this group and appreciated the time we had together so much. So I just want to, I hear his voice through these meetings and see his face and everybody here. So I'm sending love from his family to everyone and appreciation, and thank you for participating in a wonderful day. I don't know, Carol, if you are the boss here, so do you want to close us out?
No, you guys have done a great job. Thanks so much for putting this together. This was a fantastic specialty day. I just really thought it was fantastic. So thanks to you, Pietro and David, for pulling this together. And I sent a chat out. Thank you to our sponsors, our industry sponsors, for helping us to pull this off as well. And thank you for all the MSTS for attending and your engagement. This was just really, really wonderful. It's great to get together virtually and hopefully in person in October. So thank you.
Thank you, Carol. Bye, everybody.
Bye, everybody.
Yeah,